Welcome, everyone. I'm Jeff Pettis. I'm president of Apimondia. I'm also part of the BeeGuards Consortium in Europe. And you're tuning in to a Tropilaelaps webinar, which unfortunately got canceled last time because of some technical issues, but we're really glad that you're back. And we're really glad to be sharing this information with you about an emerging pest. So the reason you've tuned in is probably because, as you know, Tropilaelaps have been found in Europe and Eastern Europe, and seems to be on the move. We've gathered a panel of experts which are going to share much of the information that they've been working on over the years. And again, this is for information. I'll remind you that this will be recorded, so you don't have to, lock in on every minute. You're going to get a lot of information. But I'm going to turn it over to Raquel, our co-host, who's going to talk to you about some of the issues of the webinar itself. We're going to actually, Raquel, please, I'll turn it over to you.
Hi, good morning, everyone, or good evening, or good afternoon. Thank you for tuning in for the webinar. It's happening. And so just some general announcements that I also popped in the chat. If you would like some translation, you can just click the button, the CC button for translation and captions at the very bottom of the Zoom menu on your screen. Also, you can just submit your written questions on the topics on the chat, and Jeff will be filtering them out and categorizing them. And if you find some proper questions, some relevant, please upvote them, because we'll be prioritizing them, counting the number of upvotes that indicates some relevance. And then at the very end, in the Q&A, the speakers will address most of the upvoted questions and answer to you, and we'll have some more engagement at the very end. At the very end, we'll also have some circulate a web feedback form that will be emailed to you as well. And as Jeff said, we will be recording, and this will be made available on our social media channels later on this month or early next. So I hope you enjoy it, and we'll have some poll questions now, so we can grasp whereabouts you are from. And so please do answer a question so we understand more of our audience. And thank you.
Yeah, so if you would just take a moment and answer. It's only 4 simple questions. And again, we like to see the diversity of audience that we're reaching out there. So if you just click through and answer the questions and submit them, that would be very helpful. I'll just reiterate, all of the talks are pre-recorded because that way it's seamless. We don't have to worry about transitioning between the different speakers that you see, Giles Budge from the UK, or Cecilia Costa from Italy, or Alex from China. We've got speakers from all over the globe. So if we could run into many technical issues if we had them each start their own presentation. So the presentation will run for, I think it's 1 hour and 45 minutes with all the speakers represented. There will be a 10-minute break in the middle. So if you're really dedicated and you're just locked in listening to every word, then you'll have a 10-minute break somewhere in the middle to take a bio break. And again, use the chat to submit your questions. And if it's a question that you like or you want definitely answered, then make sure you recognize or upload it. And I think that may be it. We're again at the end, it'll be a live Q&A. So we'll have almost all the authors who have presented, have them online live, and we'll try to answer as many of the questions that you have during that time period. So that'll be the interactive part of it. You're going to get a lot of information, Watch for some of the citations in the slides, because you may want to jot them down. There's been quite a bit of research done. Not enough. We're still doing a lot of research, but there is quite a bit of research that's already been done, and you can benefit by going to those papers, finding those papers online and reading them for more detail. What we're giving you here today is a synopsis of a lot of the research that's gone on. And again, you're going to hear from different people from all over the globe who have done work at that. And so, yeah. We'll continue to answer those questions. We will start the video very shortly. We're going to have a very nice introduction from Giles, who I see there is alive and well in the UK. Hi, Giles. And Cecilia Costa as well from Rome. Cecilia runs the BeeGuards program. Giles is in charge of the unit that deals with exotic pests and diseases. And so he'll make some introductory statements, and then we'll go into the science part of it. So.
Yeah, we are having 69% participation so far, and a lot of the visual inspections, it's winning right now for the method currently used, which is interesting.
So visual inspection, you'll hear a whole section on recognizing the pest and detection. I would encourage you, no matter what you do, whether you're keenly interested or only partially interested, find a video. I think you'll see it in this presentation. Find a video about Tropilaelaps mites moving in the cell and around on the cell. It's very diagnostic. Even with older eyes, such as mine, you can recognize the mite moving on the comb. Now, you can't recognize it with tons of bees walking around on the comb, but the movement of this mite is very, very diagnostic and characteristic. So I would encourage you to watch some of those videos as a way. And Cecilia and some co-authors have published a method that you'll see as well, which is a wax uncapping method, which I think is very, very good, very fast and very diagnostic. So we'll hear about that. We'll hear about diagnostics. We'll hear about control. And towards the end, Jeff Williams from Auburn and myself will talk about research gaps. What else do we need to learn? There's quite a bit we need to learn. But again, I don't think we need to live in fear of this pest. It's very similar to Varroa. And in Korea and Thailand, where I and Jeff have worked, people still keep bees. So it's not going to be the end of beekeeping, but we certainly don't need this pest to spread. And that relies on people being vigilant. And that's what this webinar is about. So I think we're almost there. I think we're with the survey.
Okay, yeah. So I must mention, I would like to thank you so much for the technical hosting to Auburn University and Alabama Extension for the technical hosting of our webinar today, especially after our last technical difficulties. So thank you so much.
Yeah, Raquel, thank you for reminding me. We relied on Zoom last time, and many of you would have tuned in and had either 5 minutes or 15 minutes of frustration trying to get on. We worked for over an hour that morning. It was morning for me to try to get Zoom to reconcile the problem. It never happened. So we understand that frustration, but we're glad that you're here with us now. And yes, Auburn University has kindly allowed us to use their Zoom platform, and it's working, and we're delighted to be with you. And thank you for taking the time and interest in this webinar. So I'd love to get to 80% participated, but I think that's I think we're pretty good. I think we can stop with the survey. In my opinion, I think we've gotten a good feedback from our audience online. If everyone's in agreement, do you think?
Jeff, yep, we're all good. Polls
Stop the survey.
Yeah.
Okay, we'll now move on then to the main webinar content.
Yeah.
Hello, ladies and gentlemen. I am Cecilia Costa from the Apidology Group of CREA, the Italian Agricultural Research Council. This webinar is organized within the BeeGuards project, which I have the honor of coordinating and which is funded by the EU program Horizon Europe. The BeeGuards is a diverse group of actors. There are 27 of us from 16 different countries in Europe and beyond. Among the partners, there is Apimondia, who is organizing this webinar today, and also several other of the partners who are here presenting. The aim of BeeGuards is to promote resilient beekeeping across multiple levels by adapting beekeeping management practices, by furthering our understanding of biological mechanisms involved in bee health, and by mitigating environmental impacts. Among the specific aims of BeeGuards, Two are particularly relevant for this webinar today. One is colony management with low chemical input, and the other is supporting the preparedness of the beekeeping sector to emerging parasites. In this figure, the BeeGuards structure is represented. I want to underline that at the heart of BeeGuards is an innovative management system which is based on brood interruption for Varroa control. But we will see in the course of this webinar how relevant and important this kind of management can be in the face of the prevention and control of Tropilaelaps mercedese. The BeeGuards project is testing the innovative management approach in its institutional apiaries managed by BeeGuards partners across several different countries. However, we also wish to involve beekeepers in testing and validating the methods. And this is why we have recently launched a citizen science initiative with a simplified protocol which can be adopted at various different levels of participation. And if you are interested, you can scan the QR code in the top right corner and fill in the form and sign up to the initiative, which also includes a part on pollinator competition if you are interested in this aspect. And for further information, you can check out our BeeGuards website and also have a look at our social media or listen to our podcasts. I now pass the microphone to Giles Budge, who is leading the BeeGuards work package on parasites and pathogens. And I hope you all enjoy the webinar.
Thank you, Cecilia. And thanks to everyone for joining us today. My name's Giles and I'm the work package leader for work package 6, which is all about parasites and pathogens. And like all other elements of BeeGuards, it's a very collaborative affair with 8 different partners contributing to this work package. So work package 6 is all about trying to better understand how abiotic drivers like management and climate and nutrition can contribute to the severity and the risk from parasites and pathogens and the diseases that those pathogens cause. And we're going to be working on Varroa and better understanding how Varroa populations might respond to climate change, but also looking at different diseases across Europe and seeing how they might fluctuate in the future. We're also setting up a system for bee breeders across Europe to capture breeding values when it comes to resistance to some of these diseases to help mitigate future problems. But the focus of today is all about invasive pests, and you can see on here 2 small hive beetle, which we're not going to talk much about, but we also see Tropilaelaps on here, and we're really interested in trying to raise awareness and improve our preparedness across Europe for this invasive mite. An important part of our project is trying to raise awareness across the beekeeping community, not just in Europe, but also to other parts of the world. And one of the ways that we've been trying to do that is by sharing video footage and photography footage from ourselves, but also from our colleagues in the UK, which really show how the mites move. They don't move like Varroa mites, and in some ways they don't really look much like a mite at all, because their front set of legs, they move very much like antennae, so you might mistake them for a small beetle. These videos are available online if you follow the QR links, and we encourage you to go and look at those in your own time. We're also trying to highlight the size differences between Varroa and Tropilaelaps mites. They're about 1/3 of the size, and it's important that we help beekeepers get their eye in when it comes to spotting this invasive mite early. We've also made these awareness leaflets for Tropilaelaps mites, which have been produced in 8 different languages currently. But if you need a different language, then please get in touch, because we're very happy to make more languages available for beekeepers. And again, this is about trying to raise awareness, discover the mite early in its invasion cycle, because if we do that, we're much more likely to have a positive outcome when it comes to eradication. And then the final thing I wanted to mention to you about our work package 6 is that we've been working hard to try to improve the preparedness of European beekeepers for Tropilaelaps arrival. And there are a few papers out there which talk to different laboratory and field protocols to help you to detect mites that might be present in colonies. So we encourage you again to go and seek these out. If you're interested in getting updates on the project, if you're interested in helping us to co-design future experiments, then please feel free to contact this email address and join our multi-actor forum. So that just leaves me to say thank you again for joining us and helping to raise that awareness, improve our understanding of Tropilaelaps mites. And myself and the rest of the BeeGuards team very much hope that you enjoy the rest of this Tropilaelaps webinar.
Hey everyone, Jeff Williams here from Auburn University, and for the next few minutes I'm going to highlight the life cycle of Tropilaelaps. So I'm a director of the Auburn University Bee Center, as well as an associate professor at the College of Agriculture at Auburn University, which is located in the southern part of the United States. And those of you familiar with bee culture, please tune into that magazine if you want to get monthly updates on what our lab's up to through its university series. So really the pleasure of participating in that, and that this year we really thank the editors over Bee Culture. So I've had the pleasure of working with Tropilaelaps for more than a decade now, traveling back and forth between North America or Europe, where I was situated before, and Eastern Asia. So I first worked with Tropilaelaps in the lab of Panwan Chantawanakul. She was a professor at Chiang Mai University, worked very closely with our postdoc, who's now a lecturer at Mae Fah Luang in northern Thailand. His name is Professor Golf. And more recently, I'm working with Professor Baum. She's working at Chiang Mai University, also in the Faculty of Agriculture. And this past summer, myself and one of my postdocs, Dan Arell, visited Chu Lui Yung in Andong National University. So really great connections over the years with Asian partners. And that's really allowed us to hopefully build a solid knowledge base about Tropilaelaps. So when we talk about Tropilaelaps, we're really focused in on one species, Tropilaelaps mercedes-a. And so that's the species of primary economic damage to Apis mellifera, and it seems to be spreading westward into Eastern Europe. And we'll hear more about that later in this seminar series. So I'm really focusing on Tropilaelaps mercedes-a right now. Its native host is Apis dorsata, but most of our information is coming from Apis mellifera just because it's mostly well studied, easily managed, and we can really just gain access to those colonies to investigate aspects of Tropilaelaps mercedes-a parasitism. When reflecting on the life cycle of Tropilaelaps mercedes-a, it's really easy for us to first think of how it compares to Varroa destructor, that deadly parasitic mite that globally is inflicting disease globally in our Western honeybees. And first and foremost, both of them are brood parasites. They both feed and reproduce along developing bee or bee brood. They feed on hemolymph or fat body, and they can also vector viruses, which really, again, inflict these economic penalties. So there are some major similarities. Again, both are brood parasites, but there are also subtle differences in their life cycle and biology that we hope that we can kind of tune into, particularly when we're thinking about managing Tropilaelaps mercedes-a. So Tropilaelaps mercedes undergoes 5 major development stages, starting from the egg, larva, to nymphal stages, and then to the mature adults. And the life cycle really starts out when a mature mated female enters a partially capped or just about to be capped bee brood cell containing late stage larvae. And at least in Apis mellifera, there seems to be a slight preference for drone brood. The authors of this study think that it's more due to the aggregation of drone brood in Apis mellifera compared to Apis dorsata. So there might be a slight preference there, but a lot of other studies have highlighted really no difference between infestation of worker and drone brood. So perhaps not an obvious management action like Varroa destructor moving forward. So once the mated female moves into that cell, it will start laying eggs during the bee's pre-pupal bee phase. So we've seen evidence that the eggs are laid within 1 to 2 days, and those cells may still have spinning larvae that are capped over. But we've also seen evidence that the eggs may be laid as late as 2-day-old pupa. So that's really like 6 days after kind of cell invasion. Eggs are laid about every 24 hours, which is a little bit quicker than Varroa destructor eggs are laid. And another key difference here is that the Tropilaelaps mercedes females tend to lay daughter eggs first. And so if they are laying multiple eggs, they'll first lay 1 to 2 daughter eggs, followed by male eggs after this. And of course, Varroa will lay male eggs first. So it takes Tropilaelaps 6 to 9 days to develop from egg to mature mite. Also some evidence to highlight that males may take slightly less, maybe up to 24 hours less than their female counterparts, but roughly about a week time for that egg to develop into mature. So plenty of time within that like 12-day cap brood cycle for worker Apis mellifera. By the time the bee emerges from its cells, we only find either mature mites, males or females, or deutonymphs, which suggests again that these Tropilaelaps females that are infesting cells are stopping egg laying around that 2nd-day-old pupal stage of the bee. Typically, there's about 1.5 progeny per mated females that are entering these cells. So sometimes males may not even be produced. And we do think that any males that are produced would be mating with their sisters within those cells. So the majority of the mating occurrences are happening within brood cells, but it's possible that they could be mating outside those cells as well. So some evidence within brood cell mating. Those mature mites, as they leave the cell alongside the developing bee, will live for about 50 days if they're females, but only 5 days if they're males. So there's several opportunities for reproductive cycles within female mites, knowing that they're within that capped worker brood cells, for example, for maybe 12 days. So they may have 3 or 4 reproductive cycles. And another major difference is that those females, when they're emerging, they don't require any kind of like feeding, particularly on adult bees like Varroa destructors. So they can really rapidly emerge from the cell and jump back into new brood cells to infest those late stage larvae. And we think that the Tropilaelaps females will only be outside of brood cells for about 1.5 days. Again, really a critical difference compared to Varroa that typically take about a week to go back into those cells and require a feeding meal on adults. So here's a great image from Project Apis M and a film crew called Wonderstone. They visited with us over in Thailand when we were hosting a workshop, a lot of great macro images. And so if you have a really highly infested Tropilaelaps colony, you'll often see a lot of mites crawling on frames, kind of this stop-start motion. But as I indicated in the earlier slide, they're really not on those cells walking along the comb for very long. Within about a day or two, they're going to be looking for a susceptible brood cell to parasitize. So very little time spent outside of the reproductive phase with developing bees. And so we've done a few studies. Here's me in Chiang Mai several years ago, working with Professor Golf. And yeah, we only found that the mites survive for just a couple days on adult bees. And there's been a few other studies that have corroborated this. A recent study also by Professor Golf highlighted that those Tropilaelaps mites can survive almost a week on empty comb. So maybe they're feeding on something, some kind of detritus. Maybe there was some pollen or nectar within the cells. So there's a lot of question marks still over thinking about alternative hosts, but also alternative food sources, just to see what's driving the survival of mites away from brood. And this is really, really important as we think about like the distribution and susceptible areas that could be invaded by Tropilaelaps. So outside of the cells, both Rogan, a PhD student at the Auburn Bee Center, as well as Dan Arell, our postdoc, highlighted that female mites are exiting on workers as they're departing the colonies. So clearly there's natural ways of dispersal within an apiary and within the environment. So this is also pretty critical to better understand this, particularly in areas where the mites are invading. So that article was recently published by Rogan, and there's another follow-up article coming out with Dan pretty soon here. So just in summary, again, really nice to compare Varroa destructor that we're familiar with Tropilaelaps mercedizae. Both are parasitic mites of Apis mellifera, reproduce alongside brood, vector viruses. Tropilaelaps mercedizae does not feed on adults as far as we know, and is only really on adults outside of brood cells for about 1, 2, or 3 days before they re-invade the cells. They don't need to feed on adults, so they can start laying eggs really rapidly. And their egg laying interval is a little bit quicker than Varroa destructor, as well as at least one study from northern Thailand highlighted that they have a little bit less non-reproductive rate. So all in all, it seems like the population levels of Tropilaelaps should be able to increase a lot rapidly compared to Varroa destructor under the proper conditions. And sure enough, we've seen some evidence of that. Dan has been working with some data that we've collected in southern South Korea as well as northern Thailand that highlight that Tropilaelaps population can really increase, like a 10-fold increase over 2 months versus a 2-fold increase over 2 months in that same location, same colonies. In Korea and Thailand seem to be even quicker, 20-fold increase in Tropilaelaps populations that are managed over the course of two months. So really lots of opportunities for high degrees of parasitism of honeybees when they're not properly managed. So stay tuned for that. Dan's still crunching the numbers. Yeah, with high population levels comes high levels of disease and symptoms, can harm developing bees, as we can see here, a lot of uncapped cells, diseased brood, and they can also harm bees that do reach adulthood. So here's a paper highlighting overt signs of deformed wing virus that myself and Professor Golf did several years ago. So in North America, we are taking quite a bit of preemptive actions. We've got some great resources. You can visit that website, aub.ie/tropy, or scan that QR code to take you to a resource page with some information, really great photos and videos that were sponsored by Project Apis M and the Wonderstone film crew, and a lot of awesome macro photography that are really awesome resources. And then also an up-to-date distribution map of Tropilaelaps mercedizae associated with Apis mellifera. So check out that website for more resources, and really want to give a great shout out to Rogan and Dan. They've really been the driving forces of our Tropilaelaps work at Auburn over the last couple of years. And then we've had some great partnerships with Professor Baum at Chiang Mai University and Professor Chu Lui in Andong National University. And also really great thanks to all of our project supporters in North America, and really that foresight to help us understand what's going on with Tropilaelaps so we can better prepare for it. So working closely with the Apiary Inspectors of America, APHIS, as well as ARS at the USDA, Project Apis M, Pollinator Partnership, Honeybee Health Coalition, National Honey Board, and then 2 state Department of Agricultures, the home state of us in Alabama, as well as the North Dakota Department of Agriculture. So thanks to everyone for making our work possible. So thank you very much for your time.
Hi, I'm Rogan Tokash in the Auburn Honeybee Lab, and today I'll be talking about Tropilaelaps and its current distribution. So I'm currently a PhD candidate under Dr. Jeffrey Williams, and most of my work with Tropilaelaps has been involved by going over to Thailand for multiple trips and working with Dr. Barjari Chatong at Chiang Mai University in the Marl Honeybee Lab in Chiang Mai, Thailand. I think when we're first talking about Tropilaelaps, I think it's important to note that Apis dorsata and Apis laboriosa, giant honeybee species, are believed to be the original hosts of Tropilaelaps. And it's believed to be that because Tropilaelaps mite distribution historically has overlapped with the giant honeybees, and this really implies that these mites might be co-evolved together. So if we look at this figure, this is showing just the general distribution of those giant honeybee species. Now we know that there are 4 species of Tropilaelaps, but only 2 of them are capable of parasitizing Apis mellifera colonies, Tropilaelaps clariae and Tropilaelaps mercedisae. And of those 2, Tropilaelaps is the main species that we're interested in. And the reason that we're mainly interested in them is because only Tropilaelaps mercedisae has been found significantly outside the giant honeybee's native range. So this is from a publication 10 years ago, which was showing the distribution of where these different Tropilaelaps species were found. And if we overlay that distribution of the giant honeybee species, we can see that Tropilaelaps mercedisae is really the only species that has a lot of dark gray outside that native range of their host species, the giant honeybees. So here I wanted to create a map of the most current up-to-date information with Tropilaelaps mercedisae associated with Apis mellifera. And one of the reasons that I wanted to do this is that overall there's conflicting information on where Tropilaelaps mercedisae is currently distributed based on the different publications and different reports. Certain publications and reports say that Tropilaelaps is in different countries that might not be true. And so I wanted to create a map with the most conservative up-to-date information. On this map, it only includes Tropilaelaps mercedisae associated with being found in or on Apis mellifera colonies. And it only takes peer-reviewed publications where they morphologically and or molecularly identified that Tropilaelaps mercedisae mite. And so what I want to do again is overlay that giant honeybee native distribution. And you see that there are findings some countries where Tropilaelaps mercedisae. And some of the interesting things is that there's been recent detections in more Western Asia and Eastern Europe with these different publications that all came out in the past year. And so this is really important to show that Tropilaelaps has been able to expand its geographical range even further. Now, with this map, I also want to caveat, and I think it's important to show that there are additional confirmations of Tropilaelaps mercedisae in some of these countries. Places like Pakistan, Laos, Sri Lanka, and Malaysia have all had Tropilaelaps mercedisae confirmed in these countries, but they've been confirmed with hosts being the host species of Apis dorsata. So it's important to note that likely there are Tropilaelaps mercedisae mites parasitizing Apis mellifera bees in these different countries, but we don't have the actual data to support that as showing this on the map. Now, as one last thing that I want to point out with this map is that we have some large gaps that need to be filled regarding where Tropilaelaps is currently distributed. If you look at a country like, say, Cambodia, you would assume that since it's so close to Thailand, Thailand has had Tropilaelaps mites for an extended period of time, you likely have Tropilaelaps parasitizing Apis mellifera bees in this region, but we don't have any reports on that. And then if you look, we still have quite a bit of open space and a lot of gray area in between our new detections compared to our original detections many years ago. And so we're really trying to work with different researchers in these regions to really be able to come up with faster monitoring methods that are going to be able to let them do that at a larger scale and be able to hopefully fill in some of these gaps that we have in our current map. One of the ways that we did that is that one of my my postdoc colleagues, Dr. Dan Arel, recently went over to Azerbaijan because they were worried that they potentially had detections of Tropilaelaps mercedisae. And so he gave multiple lectures on how to properly monitor and detect for Tropilaelaps, as well as how to potentially manage it in the future. I also want to point out that Tropilaelaps mercedisae is one of the 6 reportable bee diseases on the World Organization for Animal Health. Now, unfortunately, while it is reportable, there's only one report as of March 2025, and that was from the Republic of Georgia. And so it's really important that people begin reporting Tropilaelaps mercedisae because this can be a really standardization and allow us to know where Tropilaelaps is at in some of these regions. If we continue to rely solely on peer-reviewed publications, we have to deal with that peer review process. And so this process might be able to allow us to more rapidly understand where Tropilaelaps has been detected. For the continuing efforts to provide current information regarding the confirmed presence, I'm looking at taking the information that I gathered to create this table and turning it into a short communication to dispel some of the other publications that have differing reports on where Tropilaelaps mercedisae has been located. And I also want to point out that we have some really good resources available from Auburn University and the Apiary Inspectors of America on the honeybeepest.org/tropy. This is where we have our current map, and then we're also looking to update that map as we go, as we continue to see more reports of Tropilaelaps being reported in these different areas. And so we'll concurrently and constantly update that distribution. With that, I'd like to thank you all for your time, and I'd be happy to take any questions at the end on the panel. Thank you so much.
Hello, everyone. Today, I will be talking to you about the range expansion of Tropilaelaps mercedes. This talk was prepared by Anna Brandorf and myself, Victoria Soroka. I will start by showing you the world map in which you can see in yellow the original site of Tropilaelaps mercedes, where it was originally present and established, and the striped area, which is the area in which, in some of the sites, we already found Tropilaelaps mercedes. If you look at the left side of the slide, you can see that there were some places in Russian Federation that we found Tropilaelaps already 4 years ago in 2021. There are also areas in Georgia where the finding of Tropilaelaps was reported in 2024, and also in Uzbekistan in 2023, and recently in Kazakhstan in bee packages in 2024. Now, if we will look specifically where all these places are, you can see that the yellow spots show, I would say, findings up to 2024, 2023, and this means South Korea and China and Uzbekistan. And here we see some kind of continuation, but if we go in red, we can see all these spots of detection of Tropilaelaps, which are quite grouped, and which might suggest that population there is not so new. And as I told you before, it was already found in one of the places in Krasnodar already in 2021. More recently, we also found Tropilaelaps in Timen and in 2 places in Kazakhstan, as you said, in Shymkent and Urjar, which is quite remote. And we believe that all these 3 places were a result of quite recent introduction in bee packages. Down below, you can see some places which I marked by question mark, which indicates this area which is really bordering those infested area and might be suspected and should be suspected, I would say, but we don't have any evidence yet if any Tropilaelaps was detected there. Now the question, are all these populations just new populations, or maybe they are already established and they are changing? We don't. So for this reason, we looked at comparison between different populations of Tropilaelaps. In all these Russia, Abkhazia, Georgia, Kazakhstan, China, and India, we compared the size, the body size, the length, and the width, and we didn't see any dramatic differences, not between our population, and it also looks that it's not really much different from those found in India by Haran et al. 2021. When we are looking at the paper by Mohammed Zenamin in 2024, which compared Uzbek and South Korean population, they found some morphological differences in Kalicera, but the significance of these differences, I mean physiological, biological, are not yet clear. And I think we should keep on looking because those changes or additional changes in established population may have biological and physiological significance for the establishment of those individuals and populations. So now that it is clear that Tropilaelaps is moving on, moving north and moving west, it is very important to understand the process. In the general process is presented in this curve, and this is invasion curve, which is actually was generated by colleagues that worked on other invasive species, plants and pollinators, and other invasive pests. And as you can see, it has this exponential curve, which starts somewhere in introduction, accidental introduction usually. And then quite after some time, there is a detection. And then quite late, there is public awareness. And it typically begins very, very late. And if at that time of detection in this area, you can prevent and eradicate the population, later on, when usually people become aware of it, the effort are immense, but the outcome is really poor because to eradicate, it's very problematic. So as it progresses, it goes more and more towards establishment of new population. And then we are going to local control and management, which are very problematic. And we're very well aware that up to now, we are still fighting with Varroa mite that invaded. I would not say the same way, but very similar way. And we are still cannot say that we are controlling the situation. So if we are looking at this curve and at those important 3 points of introduction, detection, and the awareness, these are the most important points. So now let us look at the situation of Tropilaelaps. And in this slide, you can see when you can take out the comb and really see the Tropilaelaps are running out on the combs. And in this case, as far as our experience goes, this occurs when about 40% of brood cells are already infested. So to go by this method is quite too late. I have to point out that at this point, it's not important what and how, who is responsible for the spread, because it looks now that it's, of course, a combination of human activity, unintentional human activity, and probably supported very much by climate changes. But the fact is the fact. There is an introduction, we believe mostly by purchasing of packaged bees from some infested areas, and most of them apparently die, but some establish and move on if the conditions are permitting it. And maybe already some of the populations already established, and they can overcome climatic conditions that were not adapted for them before. But anyway, if we are looking here, it looks like that detection and the beekeepers awareness as their timing is really the key for the success in order to limit area of infestation. So here I would like to show you how actually we can, I think, I don't think we can prevent totally the spread of Tropilaelaps, but we definitely can delay it a lot by really kind of lowering the steepness of this curve. And how can we do it? First of all, we should emphasize on education programs in order to bring beekeepers and all stakeholders awareness. So it will occur before actually the introduction is happening. Because if it will be before, and then we will use systematic monitoring, the detection will occur much earlier. This will follow possibility of eradication of suspected colonies. Of course, we should, I think, work on prevention of importation as much as possible. I have to warn you that already in the areas that we detected, we were reported that were infested with Tropilaelaps, the colony losses are really skyrocketing and getting to 70%. So going back to the curve, you see, by just changing this timing of detection, and we can, I think, dramatically reduce the spread rate. And this is, of course, the issue of really worldwide management. And here, I think that's why all of us here today. So it's time to act. Tropi is not waiting. With these words, I would like to give special thanks to our collaborators, Maria Ivoilova from Radnitsky Agricultural Center in Northeast Russia, Kirov, Orlando Yanes and Peter Neumann from Institute of Bee Health that confirmed that what was originally found in Russia is Tropilaelaps mercedes, and our colleagues from Kazakhstan, Oleg Krupsky and Uljan Nuraleva, that enabled us to see the infested colonies in Kazakhstan. Thank you all for listening. I will be happy to answer any questions. So hope and wishing us all success in controlling the Tropilaelaps.
I'm a freelance scientist, veterinarian and beekeeper from Georgia, and today I am going to discuss recent invasion of Tropilaelaps mite into my country. I think it will be useful to briefly describe location of Georgia and underline some peculiarities of the climatic zones and landscape. I think it will help to understand mite's current distribution pattern. Georgia is located at the crossroads of the European and Asian continents. It's nestled between Black and Caspian Seas and surrounded by Greater and Lesser Caucasus mountain ranges. And such a geographic complexity gives our country a vast variety of climatic and geographic zones. And the subtropical zone, which is marked on the map as a green area, seems to be ideal environment for the mite to establish its population. It's worth mentioning that during the last 2 decades, we beekeepers who are operating in this area, in Kolkhy's lowlands, have experienced quite notably milder winters, and we've felt it as our struggles to contain Varroa mite. And it seems that we don't have any more broodless state of the colonies in this region throughout the winter. Of course, there are some few weeks when colonies cease brood rearing, but it doesn't occur simultaneously across all colonies within the one apiary. Of course, if in apiary we have many colonies. And at the apiary level, we may never achieve broodless state, which is a crucial aspect to understand how Tropilaelaps mite may survive in our conditions. Regarding first case, in June 2024, I received call about suspected Tropilaelaps case from my colleague beekeeper operating in 45 kilometers from my apiary. I visited this apiary, and upon inspection of the colonies, I conducted brood sampling. Those samples were inspected by microscopy. We took Tropilaelaps mite samples, and we had DNA barcoding and morphological measurements, tried to identify it. And our findings confirmed that mite was Tropilaelaps mercedesi. In the sealed brood, we found infestation levels ranging from 1 to 32%. And this level of infestation, I mean high level of infestation, is unusual for Varroa mites in June, even though main honey flow has ended already and colonies are relatively shrunk. They have less brood compared to April and May. The highest load of Tropilaelaps mites per cell was 10, what we observed, and the higher infestation rate by Tropilaelaps mercedesi compared to Varroa destructor was also observed. And in co-infested cells, both species of mites were found to reproduce successfully. And I was fortunate to conduct this study with international team, including colleagues from Macedonian University, Chinese Academy of Agricultural Sciences, and Italian Research Center for Agricultural Environment. Regarding recognition of the problem, it was quite problematic. Official control actions have been undertaken, of course, but I am not authorized to discuss in detail how veterinarians managed this problem. What I can say to date, no press release has been made available on this topic, but notably Georgia is among a very few countries that have reported Tropilaelaps invasion. And it seems that the reluctance to report these cases to international animal health organizations immediately is influenced by past painful experiences. This includes economic consequences such as suspension of certain agricultural goods exports, and those actions have been taken after reports of animal diseases made to the World Organization of Animal Health. And this demonstrates, I think, how an issue extends beyond the beekeeping industry, like becoming a tool for economic warfare. Locally, beekeepers have been also reluctant to openly address the Tropilaelaps issue due to fears of the stigma or something else. And consequently, the extent of the current damage and potential impacts haven't been fully assessed. Discovery of the new infested zones continued during summer of 2024, and within the few weeks of initial detection, other beekeepers reached me with sad news. They have visited their apiaries and checked colonies using rapid brood decapping method, a new detection method designed by Dr. Uzunov, which occurred convenient, sensitive, and precise. And throughout the season, I maintained continuous interactions as much as it was possible with beekeepers, and was also able to gather some critical observations, which I am pleased to share with you. So, distribution of the infested colonies was largely confined to the western part of Georgia, which is expected, near to the Black Sea within Colchis lowlands, where we have mild winters. It's subtropical zone. And within the 3 months of the first detection, Tropilaelaps mite was discovered as far as in 100 kilometers from the original location it was first discovered. And suggesting that introduction may have occurred at least a year earlier and has gone unnoticed. And what was shocking for me, some colonies were found in terminal condition with all symptoms of severe infestation with Varroa destructor. And it is not usually the case for Varroa destructor to cause such damage during the early or middle of the summer. Usually Varroa destructor can cause colony collapse from September and onwards. So it was quite alerting for us. And infestation levels and severity also have been found to be significantly uneven among colonies within the apiaries, which is also a little bit different from Varroa infestation. And when I was interviewing beekeepers, appeared that infestation levels were quite influenced by biotechnological events happen to the colonies, such as swarming, making splits for them, and queen rearing, caging, and requeening. Upon discovery of the infested colonies, Georgian beekeepers acted, of course, and it's worth mentioning that some Georgian beekeepers were using queen caging by means of Varroa control measure during the last few years, and they applied the same approach to combat Tropilaelaps mite. Some of them have been using total brood removal, queen cell introduction, and all those methods were aimed to create broodless state in the colony. They also combined it with chemical treatments, and despite of all those efforts, mites were often found shortly after all control measures have been accomplished. Usually within 2-3 months, we found mites again in their apiaries, and that could be ascribed to different factors. Mite could survive some treatments, of course, but it is more realistic, I think, that mites have been reintroduced from the neighboring apiaries, because the location there mites have been found are densely populated with bees, and not all colonies are living in apiaries. Some of them are dwelling in forests, and feral colonies may act as continuous supply of Tropilaelaps mite. So in Georgian reality, I think the quarantine measures which are suggested by local and international guidelines are questionable. Their effectiveness is questionable. So key observation from 2025, in January and February, I collected sealed brood samples from the apiaries where Tropilaelaps have been detected previously. I analyzed them under microscope and found that Tropilaelaps mite was reproducing successfully in those colonies, suggesting that they don't leave colonies during the winter and they just don't disappear. I would also discuss a little bit the possible routes of distribution of the mites and the spread of the mites across the region. So we know that from 2014, mite was present in Dagestan, and the extensionist who is giving such a very interesting interview claims that some beekeepers in Dagestan practice import of the colonies from Uzbekistan, and we just use it not for the complete season, but we harvest honey, cool colonies, and buy the new splits again in spring. And this is quite common pattern among some groups of beekeepers. And since 2014, 10 years have already passed, and we had no any case in eastern part of Georgia of Tropilaelaps, and it seems that the Caucasus mountain range is quite protective, and the mite hasn't been introduced from this area, but it's quite realistic that from the Dagestan along the Caspian Sea coast, it can be introduced to Azerbaijan, if not already there. And from this road, from Azerbaijan border, it can reach eastern part of Georgia. Currently, we know that the introduction of the mite happened from Russian border along the Black Sea coast in subtropical zone, and now it's spreading to the eastern part of Georgia. I predict so, and it doesn't have any barrier also to Turkey, since we have a continuous subtropical zone across Black Sea coast in Turkey also. And the entire region, especially lowlands, will be infested within the few years. I think that from the perspective of the management of the mite, beekeepers who are living in mountainous areas and keep bees there will have less trouble to control it, since in Georgian mountains we have quite cold, severe, and long-lasting winters, certainly with some broodless periods enough to eradicate mite from the colonies. Regarding our future plans, I am happy that I am part of the international team now, and we are working on reproductive biology of the mite and behavior. So I hope that we'll be able to share some new knowledge soon about this. And I am very thankful for inviting me on the event, and we'll be happy to hear some interesting questions.
Good afternoon, folks. My name's Dan Etheridge from the National Bee Unit in the United Kingdom, and today I'm going to be talking to you about our exotic pest surveillance program and the detection methods that we're using for detecting Tropilaelaps mites throughout the United Kingdom. So the National Bee Unit is a government agency, and it falls under the umbrella of the Animal and Plant Health Agency, and it's responsible for delivering bee health programs on behalf of the UK government. So the primary work of the National Bee Unit is statutory pests and diseases. So namely, our statutory diseases are American and European foul brood, and our two exotic notifiable pests are small hive beetle and of course Tropilaelaps. And we also respond to yellow-legged Asian hornet incursions as well. So the National Bee Unit was established in the 1950s. We have around 60 field-based inspectors, and they are led by 8 senior regional bee inspectors, and I'm one of those. So we have approximately 50,000 beekeepers registered with us, and typically inspectors are going to visit somewhere between 6,000 and 7,000 apiaries per year. So it's an interesting situation. So although the Animal and Plant Health Agency is a regulatory agency, it is not mandatory to register with us at the National Bee Unit, but we do work to 2 pieces of legislation, and that's the Bees Act 1980 and the Bee Diseases and Pest Control Order 2006. So our exotic pest surveillance starts with assessing all the risk points around the UK. So you can see here a map of England and Wales, and all the exotic risk points have been given a score rating of 1 to 9. So basically, we've got everything here from freight terminals, so these are coastal freight terminals. We've got civilian airports, military airports, zoos, fruit importers, high throughput queen importers, all types of risk points located around the country, and they're all marked on this map and given a score. And essentially, the beekeepers that find themselves incidentally living around those points, then those guys are the ones that fall into the inspection program with us. So the apiaries that happen to be located around those risk point areas, we call those our nearest neighbour apiaries, and normally the inspectors would try and visit around 50 of those apiaries per year. The other 2 ways that we use our sentinel apiaries is we have a voluntary sentinel apiary, and this is where beekeepers themselves would receive extra training and tools to do this, and they would send in the samples twice a year to us. So that's something that they do with just our help and guidance. And then the other way is our enhanced sentinel apiary, which is similar to a voluntary apiary, except this time it's visited 3 times a year, and this is by our bee inspectors, and they do the sampling themselves. And that's all flagged on our automatic database called BeeBase that the inspectors use to risk assess these areas and where they're going to conduct their inspections. So in order to carry out these inspections as effectively as possible, this led us to a study that we undertook in the north of Thailand at Chiang Mai University, where we looked at 65 colonies across 6 apiaries. And the main aim of this was to strength test our standard operating procedures, but also, just as importantly, it was to come up with a robust method and a system that we were able to roll out to our bee inspectors and also to the beekeeping public. So a bit of video here of what the mites look like when the bees have been shaken off the comb. So you can see the way they move. Very fast moving, move across the comb, they skirt around the edge of the cells. And of course, in order to see these though, you would need to shake all the bees from the comb. And in poor light or with bees on the comb, you've pretty much got no chance of seeing these mites. But if you get good light, the bees are shaken off of the brood frame and you've got the light over your shoulder. This movement, this fast movement, can often catch your eye. So our standard operating procedures prior to this trip were as follows. So we would use the examination of the brood cells and comb. So, namely this would be drone brood uncapping. We'd also look at the natural floor drop. So this would be taking floor scrapings and checking the floor inserts, and also the comb bump method, which is loosely based on the work by Dr. Jeff Pettis. And method 4 here, which we didn't use in the field, but we would potentially use this in an incursion situation, and that would be using a miticide to increase the drop potential of the mites. So the key objectives then were really to strength test our standard operating procedures. Were they fit for purpose? Was Tropilaelaps detectable using these methods? And then the other thing we wanted to look at is how likely we were to pick up Tropilaelaps in the lab. Of course, the efficacy of all the field techniques combined, into one process, and is this deliverable to the beekeeping community? Are we able to send our inspectors out and do this? Are we able to train them in the detection, and would they be able to go out and deliver this in the field? Obviously, we wanted to see the Tropilaelaps in the field because that's the only way to learn. And then to add to this, which we're not talking about here, but we did do an in vitro survival study as well. So we also wanted to try some other techniques using some well-known methods that are known to beekeepers already. And we wanted one field technique to at least cover the phoretic mites. So it's just important to note here that these methods are being used for detection purposes and not quantification purposes, which is what these devices were designed to do in the field for Varroa mites. So that would be to quantify the infestation rate. So that's not the case for Tropilaelaps. We just wanted it as a detection method. So could it even detect one mite? So yes, and that was in the form of an alcohol wash, a sugar shake, and a CO2 usage as well. One of the things we noted fairly early on from the floor inserts is that when these are out in the field being used by beekeepers, the debris builds up in such a way that it can be very, very difficult to visually inspect these floors just by pulling them out and looking at them. What we would normally do is scrape these off, and these would go into be sent into the lab, and they would go through a series of endicott sieves to sieve the debris down. But even then, what we did realise is that there's all the small pieces still get through the sieves, and even then it's very, very difficult to visually detect in the laboratory. So this for us was, although usable, not ideal. So in trying the drone brood uncapping, we came across one or two issues there for the conventional use of this tool. One of which was the sort of juices and mess and exudate that's involved in this process. We found that it really wasn't that conducive to try and find a very small mite. So there's lots of bits and pieces of wax and so on, and visually while you're out in the field, not that easy to see a Tropilaelaps mite. And obviously this has been well used for Varroa over time, and as we know, Varroa very much prefer the brood of the drone. And just observationally, this isn't scientific, but observationally we've found that so far the Tropilaelaps don't seem to have any preference over drone brood. And one of the things we had to soon replace in looking for Tropilaelaps was this uncapping and the inspection of the brood. So what we did very early on was switch to this 10 by 10 100 cell rhombus down here on the right hand side. But one of the things we realised that we really needed to check in the bottom of these cells once we'd removed the brood. So we all had flashlights and headlights to shine right down in the bottom of the cells, because sometimes you could remove the brood and the mites would still remain in the bottom of the cell. And this is just a visual inspection, relatively easy to do. It takes 2 or 3 minutes to do, and it's quite easy because the mites are, you're very delicately removing the pupa or larva from the cell. And you're able to visually inspect it at the time, and then that larva or pupa would just go into a tube here and be recycled back into the hive afterwards. So onto the bump method then. We did find that we could dislodge the odd mite using this method, and we were able to perform this in the field with relative ease, although somewhat time consuming to remove the frames and shake the bees off. And of course we've got to think about using this. We're trying to formulate a framework for completing this in the field for our inspectors and our beekeeping public. And one of the things that we found is that we did, in the way that we were using this method, we were causing a little bit of damage to the brood. And this line of dead pupa and larva here, you can see this was taken from the bottom of a hive, and this would sit below where the bumped frame would sat. So although useful, we did find that in our instance it did cause a little bit of damage. So the results of this showed us that surprisingly that the sugar roll was actually quite effective for relieving the mites from the adult bees. Also the sticky floor, well, that still proved effective for drop, but as we discussed, it's not quite as easy to visually inspect out in the field, particularly when there's lots of debris on the high floors. And obviously the uncapping has still proved quite effective. So the method that we've been using for that continues, and we will uncap 100 cells of worker brood. And like I mentioned before, you're very much looking right down into the cells, because the mites will sometimes stay down in the cells. And what we did find is often that if you're blowing over the cells as well, that would often make the mites start to vacate the cell. So generally the results have helped us hit this three-pronged approach to our detection methods, which we now currently apply in the field. So this is our version of the sugar roll using the Varroa Easy Checker. So you can see here that a frame of bees is shaken to a bowl. A half scoop cup of bees is selected, so that's around 300 bees, and they are poured into the device. So that's a pre-primed device with the sugar, so it's a small amount of sugar, only less than 1/4 of a cup. The basket is then placed on top in this inverted position, and then the lid is placed on top of that. So the bees are rolled round for 30 seconds or so, and then we'll let that sit for a further 30 seconds or so before shaking vigorously into our sample bag that is labelled up with the beekeeper details. And then we let that sit again for a further minute or so before doing a final shake. The bees can then safely be released into the hive, and then that sample is sent to the lab where the sugar can then be washed through the sieves. And as you can see here, it produces a very clean sample, and in this instance we've collected a few Varroa mites, but it is a very clean sample and quite a simple way to get a sample to the lab. Just some interesting video here I wanted to share with you. You can see 3 Tropilaelaps mites there, so ignore the one in the top left, which is injured and not moving very well. But the other 2 mites you can see are very fast moving, and one in particular, the one on the right now moving across, as a much bigger mite. So this is an egg producing, a gravid female, and it's just an interesting observation that we've noticed on many occasions, the slight difference in the way that this particular mite moves and finds her way around the brood, particularly when it comes to interacting there with the Varroa mites, which is really interesting. So it's often those gravid females that are more likely to harass and run over the top of the Varroa, and they're much faster moving, which is an interesting thing to see, at least, but just an observation. So in conclusion then, we wanted to create a robust surveillance program across the UK, and as explained with our exotic risk points and the way we undertake that process, we wanted it to be engaging for the public so they could understand what it is that we're trying to do, and the research led us to formulate a simple and efficient field-based surveillance technique for daily usage for the inspectors, easily trained, that type of thing. A simple option for laboratory diagnosis, so something that could be sent into the laboratory. At least one part of the process is going to the lab. Easily trained, so that's again trained to inspectors, but also to our voluntary centre lapery beekeepers. So just really a general sort of digestible, non-invasive process for beekeepers, and that people can understand what it is and they aren't put off by anything that we're doing during that process. So thank you for listening to the talk, and I hope that gives you some insight into what we do in the United Kingdom. And thank you for all our support, particularly to the team and of course to Dr. Badri Chutong for her support from Chiang Mai University, and of course Bee Disease Insurance Limited for their continued support from the start on this project. Thank you very much.
Hi, I am Alex Uzunov from St. Cyril and Methodius University in Macedonia and the Bee Institute at the Chinese Academy of Agricultural Sciences in Beijing. Honeybees, beekeeping and breeding have been my most important subjects and fun for almost 30 years, but unfortunately they are constantly challenged by parasites like Varroa and recently Tropilaelaps. As well known and already described, Tropilaelaps mercedese is an ectoparasite on the Asian giant honeybees, but it also affects the managed and economically important Apis mellifera or the western honeybees. So, applying an efficient detection method for this mite is essential for the concerned beekeepers. Then presented the known methods and highlighted their advantages and disadvantages, and it seems with an overall conclusion that they are either time-consuming, ineffective, or harmful to the honeybee colonies. Now I will introduce you to the rapid brood decamping method, a new technique developed to detect and monitor Tropilaelaps mites under lab and beekeeping conditions, and a method that to good extent overcomes the difficulties of the other methods. The method was initially published as preprint, but has recently been accepted for publication in Apidology. So the method is based on the specific behavioral and biological features of the Tropilaelaps mite. And in fact, I was inspired by reading Maggie Gill's articles, and in particular her article in Bee Culture, where she reported that gently blowing over the open brood, in her case using forceps, makes Tropilaelaps to emerge from the cell and to run over the comb. However, at least for me, using forceps to open many brood cells, it was too slow and not an option under field conditions. So during my work at the German Bee Institute in Kirchhain, I had a chance to learn an interesting technique developed by Adrian Sicanu, scientist from Romania, who used fabrics immersed in hot wax to open multiple brood cells. Then in Kirchhain, we used commercial wax depilation strips as a more simplified solution. In both cases, the method was applied to control or to study Varroa mite. Then we did the testing of the wax strips and the procedure on the colonies at the rooftop of our institute in Beijing, and then in parallel with Dr. Irakli Janashia from Georgia, where we already reported for Tropilaelaps presence, we organized the study and tested the entire method. The method works on a sealed worker brood with an idea of decamping multiple cells and making mites emerge on the surface, but without removing the pupa out. Concerning the equipment and tools, you need basic stuff such as forceps or even toothpicks, then wax strips, a sticker to identify the colony if you check more colonies, a bit of paper or a cotton ball, and finally a camera or a mobile phone with a holder if you want to record the process. Once you place the frame on the table, the first step is to check the age of at least 3 sealed brood cells to identify the right brood area. That helps you to better notice the moving mites right after the decamping. Then warm up the wax strip with your hands to better stick to the brood cappings and apply the strip on the brood area, which usually covers around 250 worker cells. Press the strip against the brood cells gently to enable good contact, but also to avoid brood damage. Next, start the recording if you use a camera and rapidly remove the strip. The mites emerge on the comb surface, they move around and are visible, but then it seems that they slow down after 10 to 15 seconds, which makes them hard to notice. The mites are visible even with the naked eye, but you must keep in mind the field conditions. If you want to detect the presence, you may go without recording, but for more detailed observations and data collection, you need to use video. In the lab, we checked 28 samples with around 200 worker cells from each comb site. Of 28 samples, 11 or almost 40% were positive on Tropilaelaps, of which 2 were only Tropilaelaps infested and 9 were co-infested with Varroa. In these samples, we found only 1 cell that was co-infested with Tropilaelaps and Varroa. Now, of those 11 positive samples, when we checked the video recordings, we confirmed for 10, which means above 90% detection efficacy using video. So this is in fact the most important aspect or benefit of using rapid brood decaping. We also asked different observers to check the same videos and found that all 7 persons identified the positive samples, and plus there was no significant statistical difference between the number of mites they reported. On top of that, the observers on average needed 20 seconds to detect the final number of mites. That is why we suggested shortening the recording interval from 60 to 30 seconds. When in the lab we compared the number of mites observed in the video with the number of remaining mites in the same decapped cells, we found a high significant positive correlation of 0.9. Then the correlation of the video observed mites with the mites of the backside of the same comb sample was even higher, 0.96, which really demonstrates the method's reliability. Next, we addressed the beekeepers' concerns about the brood damage after applying the method. So in Georgia, Italy and Croatia, we tested the brood survival rate on 29 colonies 24 hours after the depilation or the decaping, and found that almost 94% of the brood cells were again sealed without visible significant damage. Now to sum up, rapid brood decaping is characterized by high detection efficacy. As the name says, it is rapid and fast. It can be used in early infestation phase. It is not expensive, and finally it is bee-friendly, which means high survival rate of the inspected brood. In contrast, the method requires good preparation or some practice or training. The first feedback was positive, and most importantly, the beekeepers, for instance from Georgia and Turkey, already started using it. Plus, on our website tropilaelaps.info, we saw increased traffic and interest in the instructional video clips that are already translated into 11 different languages. However, now as the next step, we aim to improve the method by using image processing and computer vision techniques that hopefully will allow mites auto-detection and even estimation of the mites number. This study is ongoing and it is organized in cooperation with the colleagues from my university. Our goal is to develop a smartphone application that will be available to the beekeepers. Okay, the work was done with my colleagues from Georgia, Italy, China, Croatia, Turkey and Macedonia as part of the BeeGuards project that now de facto guards the gates of the continent from the mite. The project is funded by Horizon Europe and implemented under the leadership of Dr. Cecilia Costa, who already presented the project's action and aims. At the end, I hope you find all this information useful. Thank you very much for your attention.
Hi, today I'll be talking about acaricide trials for control of Tropilaelaps mites. I'm Dan Orell. I'm a postdoctoral research fellow at Auburn University, and in the past year I've had opportunities to do Tropilaelaps research in Thailand and in South Korea. Here are some pictures of me assessing sticky boards and capped brood for mites, these little Tropilaelaps mercedisae mites, like you see in this picture on the bee's leg. I've got a few things on the agenda today: key facts, some theory, summary of what we know to this point, next steps, and then wrapping things up. So to start with, there are two key facts that I'd like you to know about Tropilaelaps mites. First of all, Tropilaelaps spends most of its life cycle in capped brood cells, like in the bottom picture. That means that for much of its life cycle, it's physically protected from treatments. And the second fact is that among acaricide options, formic acid is the option that has the strongest evidence for being an effective Tropilaelaps treatment. Into some theory. I'm going to go back to some key differences in biology that Jeff Williams mentioned. Varroa, when you compare to Tropilaelaps, has a longer dispersal phase, that is, a longer phase outside of those capped brood cells. And this is partly because the newly emerged female Varroa require a feeding period on adult bees to have optimal reproduction. Tropilaelaps mites do not require this feeding on adult bees, and they spend only about 1 to 2 days outside of brood cells, meaning that they can reproduce faster, and because they go back into those cells, they're harder to target with acaricides. For acaricides to work, what needs to happen? First of all, the toxic substances must reach an organism to cause toxicity. And then second of all, we need to be using chemicals that are toxic to our organism of interest here, Tropilaelaps mercedisae mostly, but there are 4 Tropilaelaps species. And there's quite limited information about the toxicity of different chemicals to Tropilaelaps mites. So, for example, the LD50 as a measure of toxicity. And LD50 is affected by many processes that differ between species, so details of how well they can absorb chemicals, how they move through the body, if they're metabolized in detoxifying ways, excreted or stored, as well as just how susceptible the target organs of pest are. And then thinking a little more about how are acaricides actually getting to the mites. And this is a little bit in the realm of speculation, but I'm going to speculate that if you put a certain contact acaricide into the hive, Tropilaelaps mites may be exposed to lower doses than Varroa mites. And that's because they have a shorter period outside the capped cells where they can effectively get those residues, here marked in blue. But the second thing is that a big part of how contact acaricides are spread through the hive is by bee-to-bee contact. So here I'm, at least in my mental model, is that for a lot of contact acaricides, the bees would have a higher concentration than other surfaces in the hive during the treatment period. And because Tropilaelaps mites tend to, when they exit the cell that they developed in, they tend to travel on the comb surface to get to their next receptive cell. Instead of, what they rarely do is to have a phoretic period on bees and then try to get to a next cell to infest. So I'm hypothesizing that by taking that comb route, they experience less residues. On the plus side, I think there are reasons to think that Tropilaelaps might be more susceptible to some toxicants. On this side-by-side picture, you can see how much lighter colored the Tropilaelaps mite is. So it appears that they have a thinner, less sclerotized exoskeleton, and the thickness of the cuticle and its sclerotization, hardening, can affect the toxicant uptake. When it actually comes to what do we know as of now kills Tropilaelaps mites, I'll go through that. What we want to see is these very hard to see small Tropilaelaps mites on sticky boards or on the hive floor. Starting with the most promising options, formic acid, and brood breaks. Brood breaks are non-chemical, but these are the two most well-supported options. Moderately effective, we place the synthetic chemicals flumethrin, fluvalinate, coumaphos in there, as well as sulfur. And then in the not effective category, we consider amitraz, hops beta acids, and oxalic acid not effective against Tropilaelaps. I'm highlighting amitraz and oxalic acid red because these chemicals are used widely to control Varroa, and beekeepers should not expect that if you reach for amitraz or oxalic acid that it'll be an effective tool against Tropilaelaps. I'll go through some acaricides that have been tested against Tropilaelaps. It includes synthetic, natural acaricides, and then cultural control. And there's been limited testing of oxalic acid and thymol. That's a bit of a gap in the research thus far. Here I'm going to go through some studies one by one. This is a study by Jeff Pettis and a team in Thailand, where I'm just going to simply present the results at the end of a 60-day trial. Here on the y-axis, you have the infestation rate of capped brood cells. So at the end of a 60-day trial, formic acid and sulfur both had lower Tropilaelaps infestations than amitraz, untreated control, or hops acid treated colonies. There was a temporary dip from treatment with hops acid, but that disappeared towards the end of the study. And they ran a second trial as part of this broader study, and this shows that formic acid worked consistently. In the second experiment as well, formic acid ended the 60-day trial with the lowest brood infestation levels. A throwback to 1971, just very briefly, it did show some efficacy of dusted sulfur, with mite levels going from 13% down to less than 1% over 3 weeks. Another study out of Thailand tested synthetic miticides. So this is Bevarol, which is flumethrin, Czech mite, which is coumaphos, and Apistan, which is fluvalinate. And those all showed excellent control towards the end of a 6-week treatment. I'll highlight that this was in Thailand, and in 2007, quite a few years ago, we know that mites can develop resistance to synthetic acaricides. So it would remain to be seen whether these chemicals are still useful. In Papua New Guinea, so 0 here is no change from the pre-treatment mite levels. You can see that the untreated group had the highest mite levels through most of this trial. Bevarol in blue, they estimated a reduction 1 month, 2 months, 3 months, and 4 months after the trial. It was never significant, but they did estimate that it was giving some control. Next, I'll present results of a trial that Rogan ran in Thailand, Rogan Tokash, and that combined chemical and cultural control. There were 4 treatment groups, and 3 of the treatment groups received a brood break. So it was either brood break only, or brood break plus formic acid, or brood break plus an oxalic acid dribble, or a completely untreated control where the queen was allowed to keep on laying the whole time. At the start of the trial, colony strength and Tropilaelaps infestation were measured, and then the queen was caged in those 3 brood break groups for 24 days. And then on day 24, when after she'd been released and all the brood, including drone brood, had emerged, then chemical treatments were applied to those groups. At the end of, again, the 60-day trial, like the Pettis trial, colony strength and infestation were assessed. And looking at percentage of infested worker brood cells, they started really low. The control group was really high at the end, but all the groups that received a brood break had very low levels at the end, including no detected mites in the brood break plus formic acid group. Measuring mite fall on sticky boards, which is a more sensitive method. Again, you see this really high mite fall in the control group at the end, really low mite fall for all the brood break groups, but Rogan did find some mites, some Tropilaelaps mites on the sticky boards on day 60, even in the brood break plus formic acid groups. So no treatments were 100% effective, even when you combine the brood break plus treatment. And then I'm going to talk about some next steps or research gaps. One important thing to follow up on is that there are both new treatments and also understudied treatments that need to be looked at more. So synthetic chemicals, fluvalinate and flumethrin, are used widely in Thailand, as well as thymol and formic acid. So those synthetic treatments and thymol, I think, are a high priority to actually run some more studies on. And then developing novel treatments is another important avenue. I think it's also important to understand mechanistically a little better the movement of acaricides through colonies. How are these residues moving through colonies? I presented some assumptions previously about assuming that adult bees have higher miticide concentrations during a treatment, but how are these residues getting to the mites, and can we optimize delivery? It's also important to understand Tropilaelaps's intrinsic susceptibility to treatments. There are 4 different species of Tropilaelaps mites that we know of to date across a wide geographical area, and the intrinsic intrinsic susceptibility of different mite populations, or perhaps developed resistance, could vary across geography. And then to conclude, I'll leave you again with this summary of the most to least promising options. Again, formic acid and brood breaks being the most well supported. Here are my references, and I'd like to thank the funders that have made this international research possible, and I'll be happy to answer any questions. Thank you so much.
Hi, I'm Jeff Pettis, president of Apimondia. I'm very pleased to be with you today to talk about Tropilaelaps mites. My talk says that I'll talk only about cultural control, but as we'll see in just a moment, I'm going to cover a bit more in the short time that I have. So Tropilaelaps mites, cultural control. I'll touch on that experiment I did in Thailand, but I'll also touch briefly on survival on bee products and packaged bees, because we need to know about how these mites might move around the globe. And finally, I'll touch a bit on winter survival. And again, the more we know about Tropilaelaps, the more we know about how it moves, the better we are able to prevent its movement, perhaps. So just a very brief recap on the life cycle. It reproduces on bee brood, very similar to Varroa mites, but the big difference is that it doesn't need to spend time on adult bees feeding. So it spends most of its time on the comb, and that's important for sampling and everything else. All the evidence is that they cannot feed on adult bees and that they die within 4 to 9 days. And I'll touch on that when I get to the packaged bee part. So remember, very similar to Varroa, but the difference is the mite cannot feed on adult bees as far as we understand. And it spends most of its time on the comb or in the seal cell. So the research that I'm going to talk about with the cultural control was published in PLOS 1 in 2017. And the parent colonies, the undisturbed parent colonies, had higher mite group, that was the control colonies that were unmanipulated, had higher mite levels than either the parent colony or the nuke from which a parent colony was taken. So I've got large colonies. I split out a nuke from some of those colonies, and both that parent and the nuke had lower mite levels than the control, and I'll show you that data very briefly. But again, this is a simple cultural control that we can use to help reduce Tropilaelaps, and it works very similar to the way it does with Varroa. We know that making a split or a nuke reduces Varroa levels. So, yeah, the Tropilaelaps mites, they were significantly lower in both the split and the parent colony, and there's the statistics on that. The frame, you notice that frames covered with adult bees. These are small colonies. These are in Thailand, so they only have about 6 frames of bees, and the nuke obviously is a much smaller colony. But over the course of the experiment, over the 60 days, the mite levels the frames of bees were significantly different. But most importantly is this number down here. This is the number of mite-infested brood cells. And we see that by day 30, they're still very similar. But then over time, the last 30 days, the control, the unmanipulated colonies, the mite levels increase significantly. So that's the take-home message from that. Leaving the colony alone, the mite levels grew. If we simply made a split, the parent colony and the nuke contained fewer mites, significantly fewer mites. How do we measure mites, Tropilaelaps mites in colonies? Well, it's not very easy, because if you've seen some of the videos, the mites move very quickly. So what we do is we uncap, the way I do it, is we uncap 200 cells, 50 on a side from 4 different sides, and that gives us a total of 200 cells. We only report mite-infested cells. So I've uncapped these 10 cells in a row. I'll pull the content out, look in the bottom, and I just record the number of infested cells, not the number of mites, because the mites move quickly. And as I uncap, they can be moving around. So I'm actually looking at each cell as I uncap it. This is just for demonstration purposes. But so we report mites, infested cells out of the 200 that we look at. Some papers have looked at 100, and I must be a little critical here. I don't think 100 cells is sufficient, and certainly not 100 cells in one block. We move around, we do 50 on the side, and simply pick and look as we go along with light, look in the bottom of the cells, and you can see the mites on the pupae or running around on the cell. So it's very easy to do, but it is time consuming. We need some of these faster techniques, and you'll hear about some of those in this webinar. Very quickly, how long can Tropilaelaps mites survive in packaged bees? Packaged bees are used to move bees around the globe, from Australia, New Zealand to Canada, from other parts of the globe. And so this was a very simple experiment that I did in Korea, and we placed mite-infested bees in packages, placed sticky boards under those packages, and just looked at the mite drop over time from those packages. How soon did the mites die? And then at the end, I washed the remaining bees after about 8 or 9 days. When no more mites were falling, I washed the bees to make sure that there were no more mites remaining. So I tested some oxalic acid strips to see if they would speed up the process of knocking the mites off. They weren't very effective in this trial, so I won't talk much about that. What I will show is simply the mortality of the Tropilaelaps per day from these packaged bees as a percentage. So over 50% died in the first day. By the second day, we had another 28% dying. And you can see by day 4, we have just a very small percent of the mites surviving to day 4. I ran this out to 8 days and then washed, and there were no more Tropilaelaps mites present in those packages. So this is very consistent with the work from cage bees, where they say, 3 to 6 days, the mites will die without food and without access to brood. Again, very quickly, there's been 2 studies on bee products, one that I was author of, and also a group out of Switzerland and Thailand. Look at the dry pollen. Dry pollen in our study, and royal jelly and queen candy, everything died in 3 days. In the study other study out of Thailand, dry pollen 3 days. But dry comb somehow supported the survival of mites for a longer period of time. I just point this out because we don't understand what that was. They speculated perhaps humidity or something left on the comb. But regardless, so this is almost the extent to which we see mites surviving without food is 6 days. Yeah. And other studies, one study, I think, almost pushed it out to 9 days. But mites can't feed on adult bees as we understand it. And in general, they die in 3 to 6 days. Again, now very briefly, I'll talk about winter survival, because we always felt that mites wouldn't be problematic in areas that had a brood break. Well, what are areas in temperate regions that are having a brood break? I think with climate change, we're having more and more small amounts of brood reared through the colony throughout the winter. And this is some work I did in Korea. I'm just highlighting the funding sources, obviously BeeGuards, who's sponsoring this webinar, and also Project Apis in the US, funded some of this work. There's at least 3 possibilities of how mites are surviving through the winter. The first is diapause, where the insect simply, or the mite in this case, simply goes into a quiescent stage through the winter to get through and becomes dormant, basically. The other is alternative hosts, that the mite can move out of the bee colony or use other things within the bee colony or other, other alternate host, move outside of the bee colony and use alternate hosts. And the last is that there's actually brood rearing in winter. And we saw this in Korea throughout the winter. There was actually active brood rearing in January and February in Korea. So there still is brood rearing in many areas. I'm highlighting this alternate host idea because Delfinato and Baker in '61, when they reported Tropilaelaps for the very first time, they found it on dead rats and bees in the Philippines. So alternate host is certainly a possibility, but all three of these possibilities are still within the realm of possibility, shall we say. Okay. So how can we slow the spread of exotics? Beekeepers are our first line of defense. We need to educate the public. This is what this webinar is about. We definitely need better detection methods, and you'll hear about some of those during this webinar. I think sentinel hives in certain areas might be a good thing. We know that mites move on trade goods in airports or ports. And again, raising awareness. The single best thing you can do to learn how to identify Tropilaelaps is watch some of the videos. The mite movement on bee brood or moving around the comb is very characteristic. So with that, I think I'm closing and say, please come to Apimondia in 2025 in Copenhagen, and there'll be more talk about Tropilaelaps, mites, honey fraud, and et cetera, et cetera. So thank you for your attention.
Good morning, good afternoon, good evening. I am François Diaz. I work at the World Organization for Animal Health in the Preparedness and Resilience Department, where I am Scientific Coordinator for Wildlife and Bees. My presentation will be on the relevant WUA activities regarding the preparedness against Tropilaelaps. 3 main activities or tools from WUA are relevant for the preparedness against Tropilaelaps and will be detailed in my presentation. First, the World Animal Health Information System, more known under its acronym WAIS. This tool is useful for Tropilaelaps preparedness in providing information on its presence worldwide. Second, the WUA standard, useful for any countries free of Tropilaelaps to prevent its introduction in their territory. Finally, WUA expertise that can provide scientific support to countries. And I will conclude the presentation by a last slide on possible future activities that I think are necessary to complement what we have already for the preparedness against Tropilaelaps. The World Animal Health Information System, or WAIS, is a platform which allows to the WUA member countries to notify WUA on their epidemiological situation for a limited number of diseases listed by WUA. Today, we have 119 diseases listed, but the list is regularly updated. It's also a platform which allows to notify emerging diseases with significant morbidity or mortality or zoonotic potential. Regarding bee disease, currently 6 diseases are in the WUA list, including Tropilaelaps infestation of honeybees. WAIS is an essential tool to be informed on the presence of Tropilaelaps and its diffusion worldwide, and so for any country free from Tropilaelaps to take appropriate measures when importation of live bees or products of bees from other countries are done. WAIS is available online, and all the notifications from WUA member countries are public and so can be consulted on this web platform. You have on this slide the access from the WUA website and in addition the QR code to go directly on it. The WUA standards are mainly laid down in 4 publications, the 2 codes and the 2 manuals, respectively for terrestrial animals and aquatic animals. All these publications are available free online. Regarding honeybees and Tropilaelaps infestation, the relevant publications are the Terrestrial Animal Health Code and the Manual of Diagnostic Test and Vaccine for Terrestrial Animals. Let's start by the Terrestrial Animal Health Code. The aim of this publication is to assure the sanitary safety of international trade in terrestrial animals and their products, while avoiding unjustified sanitary barriers. This is achieved by specific chapters on the animal disease listed by WA and which details health measures that have to be used by veterinary authorities to prevent the introduction of the disease in the importing countries. The Terrestrial Animal Health Code is also an essential tool for supporting the mandate of the WA in the area of improving animal health and welfare worldwide. In this Terrestrial Code, the chapter 9.5 is dedicated to infestation of honeybees with Tropilaelaps spp. The Article 1 of this chapter provides important information. First, it mentions its scope. Honeybees are understood as a species of the genus Apis, and the Tropilaelaps mites are Tropilaelaps clarae, Tropilaelaps conigerum, Tropilaelaps thai, and Tropilaelaps mercedesae. Then it provides an epidemiologic information, the fact that the mite cannot survive for periods of more than 21 days away from the bee brood, and that the mite can also act as a vector for viruses of the honeybee. Finally, this Article 1 mentions how this chapter should be used and by whom. Veterinary authorities when authorizing import or transit of the commodities covered in the chapter. The general structure of this chapter and the information available is the following, as you can see it in the slide. After the general provision as described previously, you will find what are the safe commodities that can be imported, how to determine the Tropilaelaps spp status of a country or zone, and also declare a country or zone free from Tropilaelaps spp. Then you will find in this chapter recommendation for the importation of live bees, product of bees, or apicultural equipment. In these articles, you will find which technical process or treatment can be used to limit the risk of importing Tropilaelaps when importing live bees, product of bees, or apicultural equipment. This chapter and all the information presented are available on the WA website. You can see on this slide the access to the Terrestrial Code from the WA website, and in addition the QR codes to go directly on it. The Terrestrial Manual for Diagnostic Test and Vaccine is the second relevant publication with relevant standard for Tropilaelaps. The aim of the Terrestrial Manual is to provide internationally agreed diagnostic laboratory methods and requirements for the production and control of vaccine and other biological products when they exist. It's a companion volume to the Terrestrial Animal Health Code. The specific chapter addressing Tropilaelaps is the chapter 3.2.5 on infestation of honeybees with Tropilaelaps spp. You will find in this chapter all the diagnostic test method to detect, diagnose Tropilaelaps infestation. It is therefore an essential tool for the detection and confirmation of Tropilaelaps spp in any samples. This chapter is available on the WA website. You can see on this slide the access to the Terrestrial Manual from the WA website, and in addition the QR code to go directly on it. A source of expertise for WA and also for supporting WA member countries is the WA network of reference laboratories and collaborating centers. Reference laboratories have expertise on specific disease and have an expert designated. The collaborating centers have more horizontal expertise. For Tropilaelaps, we have one reference laboratory located in France with Dr. Stéphanie Franco as expert. And for the more transversal topic of beekeeping management practices and biosecurity measures in the apiculture sector, we have the Istituto Zooprofilattico Sperimentale located in Rome, Italy, with as director of this institution, Dr. Giovanni Formato. To conclude my presentation, I would like to highlight further possible activities that I think useful and needed to be prepared against an establishment of Tropilaelaps, further a preliminary introduction. It is the development of a model of national preparation plan in case of the introduction of Tropilaelaps SPP in a country free, and then to support and encourage countries to develop their own preparation plan based on this model. In complement and in addition of these, it is to encourage the countries, once this national plan has been developed, to organize simulation exercise based on this plan. Thank you for your attention.
Hey Jeff, do you want to do any intros to our little section here?
Well, sure. So I've been monitoring the questions coming in, and hopefully some of these things that we'll cover in the research gaps will come up. And to be honest, I've read Jeff's list. He has a list, but one of the first questions I got was about could Tropilaelaps invade wild bee colonies, native bees, whatever, bumblebees. And I think we don't have much knowledge on that. So we'll see where this goes, but hopefully in this research gap thing, we'll answer some of the questions that have come in. And yeah, let's just, I think there's quite a bit that we know, but quite a bit that we don't know. Right, Jeff? Let's go.
Yeah, exactly. Well, this is meant just to spark off some thought, and I guess that will flow into that Q&A. So right. Yeah, myself and Rogan and Dan brainstorm for a few minutes, just again, some talking points for all of us. And me and Jeff will go back and forth for a few minutes here, and then yeah, we'll turn over to the chat. So if a question comes up, keep writing those in, and Jeff Pettis, I think, will go through them in the next couple minutes here for all of us to discuss. So on the right there, you can see Rogan. He's got essentially a Q-tip or a swab, and so that's just a prompt for me to think about the importance of monitoring and how we need to keep working on improved monitoring methods, both from like a scientific point of view, but also from a practical beekeeper point of view. And Alex has had an awesome presentation that's produced out of BeeGuards to highlight improvements in how we can more practically monitor Tropilaelaps, but also hopefully how we can incorporate some technologies too, because we all know in the beekeeping sector, we need to start moving towards a little bit more technology, particularly when you compare us to other kind of farming sectors. So having that early detection is super important, particularly since we know that Tropilaelaps mite is about a third the size of Varroa. We've done some work over in Thailand, and also Jeff Pettis and others have kind of highlighted that at least the current Varroa monitoring schemes are okay, but not great. Here in the United States, we're frequently pushing beekeepers to do the alcohol wash. Well, we know that that's not really going to be proper for Tropilaelaps because the mite's just not hanging on to adult bees as much. One of the most sensitive methods that we've seen is the sticky board, but that takes a lot of time to look through. You really have to strain your eyes and train your eyes to see the mite. And not to mention, you have to go to your colonies twice, once to insert the sticky board and then to come back and remove it. So just those monitoring tools are, I think, something that we really need to move forward to investigate more. And then just flowing into that idea of promoting integrated pest management. So we've got proper monitoring methods. We understand damage thresholds, which right now is, I think, really like a big dark hole. I showed a picture of a frame with a lot of brood damage that I took in Thailand, and Project APISM also highlighted that through some of their videography and their videos that they have up on YouTube, and that the beekeepers there are really just monitoring for extreme damage on their colonies. They're not looking for Tropilaelaps mites. I think that's going to be another challenge, is just that whole connection to monitoring and then treating when appropriate. Jeff, any kind of comments so far?
Well, yeah, just one thing to keep in mind for the broad audience, we need to always remember to differentiate between detection, just presence or absence. We need those tools vitally in the field for beekeepers, bee inspectors, or whatever. For quantification, which is the research component that we do, if we want to evaluate how effective a miticide is, we need to really quantify how many mites are present at the beginning and at the end. So some of these methods that we've talked about, they fall into these two broad categories. And just remember that what we really need are these very fast and effective, they have to be effective detection, rapid detection field techniques. And I agree that Alex, the wax uncapping and some of these other things are moving us in that direction. Yeah.
Yeah. And just to come full circle to that image of Rogan, we've been studying if we can use eDNA as a tool. So rapidly swab bottom boards or other places in hives in a yard and send those off and hopefully get a quick response whether or not Tropilaelaps can be detected.
I had another quick thought about sticky boards. Everyone's always optimistic that sticky boards will work. First of all, as you said, Jeff, these mites are so small that as soon as you get like into the second day of debris fall, they're really hard to detect. But more importantly, we and other researchers have tried formic acid, amitraz, and all this stuff with a sticky board, thinking we'll sample the whole colony. We'll knock those mites down. They'll collect. Like I even tried it in 1 hour with formic acid. The problem is something about the way the mites die. They don't fall through the bees, or at least that's our speculation. Because I put formic acid on 1 hour, 4 hours, I'm not collecting mites or at a very high rate. So it's just not the sticky boards. To me, I mean, they can be used, but they're not so sensitive. What could be used is a detritus sample. So if you have sentinel hives on ports, you could not sticky board, but just a detritus board to collect the material and use a molecular technique, or like you talked about, eDNA, to try to find the mites in a monitoring program. But yeah, the traditional sticky board and mite fall just doesn't seem to work with Tropilaelaps.
Well, when we did our work in northern Thailand, we found that it was effective, but the sticky board has to be in for 3-4 days. In your case with formic acid, I'm wondering if the formic rapidly got a couple Tropilaelaps maybe walking on the adults, but all those, it wasn't that effective against, of course. Well, it may have killed the mites in the cells, but those cells might not have been uncapped, and the mites might not have fallen down into the sticky board. So yeah. Lots to look into for sure. And then just rolling into the preventative. Oops, sorry, Raquel, just go back one slide. Just to talk about the preventative and control options, as Jeff mentioned. There has been studies. Jeff's done some work. Others, including ourselves, like Rogan Tokash and Dan, have been working hard in Thailand and Korea with our collaborators there. So we're really trying to identify if the current Varroa products that are registered in the United States will be effective against Tropilaelaps. That's kind of like the first place to start, because that's most likely like the easiest response is just do something we're familiar with. So we've had sort of a mixed bag there. But the other thing I'm really interested in is just hearing more stories about what beekeepers in different countries, particularly in Asia, who have been living with the mite, how are they successfully or relatively successfully able to live with that Tropilaelaps mite? Because we've seen from some of Dan's data that the population growth is pretty rapid. If you don't do something quickly, there will be problems for your at least Apis mellifera.
Yeah. Very quickly, just to follow up on that, formic acid appears to work very well in a lot of contexts, but I have a question that's come in and we'll address it more. What about in hot climates? Well, that is an issue. Formic acid in hot climates is an issue. The other thing, with all apologies to Dan, he gave a very good presentation. I did very recent work in Korea, and amitraz strip, the Apivar strip, did not work. And I published on that, so that he reported that truthfully. The oxalic acid, I only used it in strip form, so we have a lot more work to do on oxalic acid. I think oxalic acid might work, but it didn't work in the form that I used it, which was a slow release strip. And lastly, the amitraz Apivar strip didn't work, but when I tried Apiflex, I think it's Apiflex, Amiflex in gel form, it did work. It worked reasonably well, not quite as well as formic. So there is promise with amitraz, and I know that some beekeepers in Asia fumigate with amitraz, which is we don't do in the US or other parts. But so amitraz still can be effective, but we need to understand how it works in the colony. Yep.
Okay, thanks, Jeff. Raquel, you come forward. There you go. So kind of back to the basics and the fundamental aspects. I mean, there's a clear need for us to understand where it is. We saw a great presentation at the end from Irakli highlighting, you know, even in a country as relatively small as Georgia, but having, you know, quite diverse landscapes, mountains, plateaus near the coast, high elevation. We really need to get a better handle on where that mite is going to be or susceptible areas. I also saw Violetta highlight the WOA database, and again, Irakli was, I think, the first. Georgia's the first country that's actually used that resource. So I think that's something that we also need to further look on and improve upon, is the reporting, the acceptability of reporting, not to be marginalized. For example, if your country has Tropilaelaps, I mean, we know roughly where it is. So there's no reason, hopefully, why other countries can't come on board with that, because it is important as this mite is spreading further west. So we've, at least one example here is a distribution map that we produced with Apiary Inspectors of America, as well as some colleagues here at Auburn. And that was part of our outreach tools that we've been working on, connected to North American bee inspectors. So we've tried to be as conservative as possible. So we really had to dive through the peer-reviewed literature, because that's the only place that is reporting it right now. And even when you look into studies, you have to really determine whether or not they did a proper morphological assessment, or did they do a proper genetic assessment, rather than just the assumption that they did a trial with Tropilaelaps mercedisae, but then there's no confirmation. And you know, we could have our hands slapped over here in Auburn also. I mean, we do all kinds of Varroa trials and just assume it's Varroa destructor without doing all of these, you know, keying to identifying or genetic tests. But I think now, particularly with Tropilaelaps, we don't really have a full understanding of its distribution. We need to be especially careful with reporting that species identification as we're doing more and more publications. So, Jeff.
Yeah, I'll just jump in very briefly on distribution. If you remember Vicky's talk, she had a lot of countries with question marks, and we hear these through the grapevine. Iran, oh, it's in Iran, it's in whatever, you know, all these other different countries. I would encourage those, as you pointed out, Jeff, to report through WOA, which is formerly OIE. I mean, we need to report it, because if we wait on the publication process, there's a huge lag time in getting publications out. But as scientists, we do have to confirm that what we have is really Tropilaelaps. There's a good example of this. It was reported 20, 30 years ago in Africa, in, I think, may have been Tanzania, I can't remember, on the old distribution maps. That was a false ID, because if it was there, it would have spread. So we do have to be aware and have the right identification, but then we need to report it as soon as possible. Yep.
Yeah. And of course, a country here the size of China or even India, I mean, there's clearly going to be differences in locations of where that mite is. So even just working more collaboratively at a global level to try to understand exactly where that mite is. And then also thinking about, you know, how it's dispersing, how it's surviving in these areas. It's a big question that we heard from Irakli in Georgia. And, you know, the same thing could be said even in South Korea. Interior parts of that country experience a pretty decent winter with a brood break, yet they still find Tropilaelaps in that country spread out. Is the mite surviving over winter through some kind of diapause or alternative host, or maybe reinfestation by migrating colonies, maybe from coastal areas or southern areas every spring and summer for pollination purposes, or reinvading the area? So lots of questions over the mite persistence, especially in kind of temperate areas, which is of course quite relevant to lots of Europe and quite relevant to lots of North America, where I'm from. So you could flip through, Raquel, one more aspect. I think this connected to distribution, thinking about habitat suitability. Right now, we think that that's really strongly connected to presence of honeybee brood, although we could be proved wrong here with some kind of alternative hosts. My colleague Golf in Mei-Fa Luang University highlighted that the Tropilaelaps mite can survive upwards of a week, even with no brood on a brood frame, empty brood frame. But yeah, how long does it take to knock off the mites? And again, what's driving its success, particularly in early spring? So one of the programs that we're running in the United States is a brood monitoring program during winter. And so here's a quick snapshot of December 19th, so mid-December, and you can still see there's several locations. The blue dots represent presence of brood, not that much, but it's still like, a handball kind of sized component of cap brood in the colonies, and it only takes a small amount of cap brood and one mated gravid female to persist and to start laying eggs again. So this is a really useful tool for Tropilaelaps suitability. We initially created it to help beekeepers identify optimal times for oxalic acid use, especially with the dribble and the vapor, because of course those methods only work when there's low to no brood. So this might be something that Europeans and other countries might be interested in monitoring too. What's going on in the winter in your colonies? And maybe Dan or Rogan can post the URL to this brood monitoring page in the chat.
Yeah, Jeff, just following up on that, part of the BeeGuards program is to actually look at that. I know you're doing a great job in the US with a lot of different collaborators across multiple states, and in Europe trying to do the same thing. Are these colonies really going broodless? That's the first question. And then if they are, then how are the mites surviving? And I just got a question in the chat about, they misread it and they said the mites can live for 60 days without brood. No, it's usually 6 days. Six days is probably, not 60, but 6 days. But again, it brings up all these things to how they're getting through winter. And we've touched on them in different ways, but we can reiterate it a bit more in the Q&A. Diapause, that the colony is not really going broodless. And then the other is that they can use alternate hosts. And I think all of those are still possible at this point. Yeah.
Hey, Raquel, I think we're almost done here. So, and the last thing, particularly connected and concerning the beekeepers here in the webinar right now, and those that'll watch this recording, is just improving awareness about the mite kind of one step at a time. So I think exactly what we're doing here with Apimondia, BeeGuards, we've been working with Apiary Inspectors of America is to try to educate our stakeholders. And in Auburn, we've started with kind of like a train the trainer program where we took a handful of bee inspectors over to Thailand so that they could firsthand see the mite. And then we hosted a larger workshop for bee inspectors across Canada and the United States to come to Auburn and to gain some information connected to the biology, but then also do some mock trials. I saw one question about, can you distribute Tropilaelaps mites like dead specimens? And we have been doing some of that here in North America with little vials with alcohol. We even did some mock alcohol washes and some mock sticky boards so that the bee inspectors can try to get a feeling for what they're dealing with and what they might see or their beekeepers might see as they're doing these inspections, particularly during the busy bee season of kind of like spring to fall. So we've got, at least in North America, lots of great resources coming up. There's 4 amazing videos produced by Project APISM and Wonderstone Films. So if you check out that link, again, I'll get Rogan and Dan to copy that in if you don't want to scan the QR code. So yeah, 4 really nice videos, lots of great macro photography, images, some handouts, and conversely, I also know that BeeGuards have got a nice website too that's got some good resources to check out. So yeah, we're slowly building up the resources for our stakeholders on kind of both sides of the pond, and hopefully in other locations like South America and Africa that don't have Tropilaelaps, Australasia, they'll also start coming online here as well.
Yeah, so there's been a lot of, part of the reason for this webinar is to raise awareness. And as part of BeeGuards, we have this flyer, front back flyer that gives some of the characteristics and has pictures and has a link to the video. So that's a nice tool. But the other thing we're planning under BeeGuards is a hands-on workshop, which would be just like you had in Thailand for the U.S. apiary inspectors. We're hoping to get apiary inspectors and other veterinarians and stuff from Europe. And this might be held in Georgia. Alex and others were working on that as possibly having a workshop so people can see it firsthand. Again, though, any of these things go a long way towards raising awareness and just educating everything from beekeepers to bee inspectors to veterinarians to be on the lookout, because information is key there. So yeah. Thanks, Jeff.
Good. And then just to wrap up, just from my side, another thing that is kind of connected to critical gaps is just the support. So far, we've had some amazing support, both from our colleagues in Asia, like Baume and Chului, but in North America, we've had some great financial support to help us do these workshops, do this research. And that really takes a lot of like foresight into trying to prevent history from repeating itself, so that we're not caught off guard with all these invasive parasites that just keep coming, Varroa, small hive beetle, we've got the hornets. So I really want to give a shout out to the USDA and some of these important nonprofits for investing in this kind of future. And again, it seems like the same thing is happening in other locations as well. So we got to keep that going.
My final thought on that, Jeff, is people think of researchers, we're all super highly competitive, which we can be, but I have to thank Auburn University for helping Dan in particular. Dan Arl, he and I worked together in Korea with Chuli Zhang, who you see the picture of there, and we've had excellent support in Thailand and other places where we want to work. And yeah, without the funding, as you can imagine, if you're a European researcher or an American researcher, to do this work in a foreign area takes a lot of logistics, and we rely on people like Chuli Zhang and Andong University or Chiang Mai University for a lot of support. So thanks to all.
Okay, turn it over.
I think we're at Q&A. Oh, yeah. So I'd like to ask all the speakers, Rogan, Giles, everyone, I'm looking around. We can all come back into the conversation. And I'd like you to think about, I've challenged you with, at the end, we're going to ask each of the speakers who are present to just give some final concluding thoughts. So we'll get to that, just things that they think are vitally important, not to really recap their talk, but to talk in general about some of the things that they think are important or where they're going with research. So I'm actually going to start with the very first question that came in. And I think we don't have very much information here, but I'd like to hear from others. And that is, can Tropilaelaps get into wild bees? The first one I think of is bumblebees. And I say that because it's a colony nesting, not so much the osmia and other small species. But do we have any evidence that it can move into wild bees? And they said, if so, can we eradicate it? Well, when you think about wild bees and trying to do a control thing, I think that's out of the question. I think the real question is, can they live in something like a bumblebee colony? And actually, that would be fairly easy to answer. Take some commercial bumblebee colonies to Thailand or Korea, or they're already there, and just try to artificially infest them. But has anyone done that? I haven't had time. But Dan, I'm looking at you. Dan, there's a research project. Does anyone have any information about wild bees or bumblebees and things like that in Tropilaelaps? I'm unaware of anything published. I see a lot of...
No, and I don't think we have information on that, but it's important, especially since there is some global trade in other bee species, like I believe the pupal stages of perhaps osmia.
Osmia, yeah. And all this commercial trade of bumblebees, sorry. This is kind of related to a question that we keep getting, which is, can they use alternate hosts? And we haven't discussed the rats or the mice, but that was in the first publication. And so far, we don't have direct evidence, but it's out there that, and it's embedded in some of the questions we've gotten. How do they survive the winter? And we've already talked about that, whether they can use alternate hosts. So does anybody want to chime in? Anyone done any work? I have not done anything on alternate hosts, but I know, it's possible. I mean, it obviously is possible. The brood levels go down. Could the mites be moving out of the colony and using alternate host? Any thoughts along that? I see Alex wants to jump in, I think. Alex, you no. No. Again, research, it's not simple because when you're dealing with vertebrate animals, there are regulations. But if you take caged mice, small mice, and you put them in cages over sticky boards and you introduce Tropilaelaps to them, it's not simple, but we could probably find out if they can in fact feed on mice. There is something that hasn't come up, and I'll give a shout out to Maggie Gill and some of her work. I was recently interacted with her, and she was using dead bees, and she thought they survived longer on dead bees than they did on newly freshly dead bees. And so there is something there that they can survive a little bit longer as the bee is decaying. Maybe they can access some food sources there. And there's this question of pollen, honey, and everything else. It's 3 to 4 days. When you get to dry comb, there's something in dry comb that it doubles the lifespan. So anyone have thoughts on that about what, again, no evidence that they directly feed on live bees, but any thoughts on other things that they could feed on? Most of the colony stuff, the honey and the pollen, they don't feed on. That's pretty clear from multiple studies. Someone be brave, chime in. Rogan, you're wanting to say something.
Well, I can just say that Dan and I did a little bit of preliminary work looking to see if alternate hosts on like wax moth larvae or pupae, they could survive on those when we were over in Thailand and in Korea, and we found that they didn't survive very well, and we think that the humidity played a pretty big role in the incubator, and we were having struggles getting them survive 2 days within the incubator. And so it is interesting to see that comb, it really was only one mite that was able to survive for 6 days, and it's interesting because we've struggled to keep them alive for just 3 days. So that does bring up a question as to how well they would be able to move to new areas.
And on that note, I also tested the survival on dark comb, and that seems it's very interesting that other group found extended survival, like 6 days on dark comb. We didn't see that, but at the same time, one of the things I think about is that nature rolls the dice a lot of times. So even if the maximum survival that we've seen in the lab on dark comb is 6 days, it seems very possible that in nature you might have varied conditions and more, essentially more mites being tested, and more days would be possible even if we haven't observed it. So a precautionary approach would be reasonable.
Right. Giles, it looked like you wanted to say something. Jump in, Giles.
Yeah, I was just going to say that I wanted to highlight an element of your evidence you mentioned earlier, Jeff, when you were talking about these mites being found on rats, dead rats, right? And then the data that we got from the first Thailand trip that I saw talked about survival that was better on dead adult bees than live adult bees. And I just think there is this sort of saprophytic nature of some of the mites from that family that we need to recognize and not fall into the trap of, like we often do, is by searching for similarities between Varroa and Tropilaelaps. Actually, the key here is the differences, the biological differences between the species. And for me, this propensity to potentially survive on dead stuff is in itself very, very interesting.
Yeah. Good point. The other thing is this group of mites in general, a lot of them are predatory, which means they don't specialize on one type of host. They can parasitize a lot of different ones. And yeah, they could be feeding on dead material. I want to bring in just very quickly, I'm being selfish. No, the package B work, almost all the mites died in 3 days. One lived to 4 days. Now you've shipped that package from Australia to Canada. It arrives in Canada, and now we've got a 6-day limit, let's say, of survival. If you shake that package into an empty comb, it still takes the queen a little bit of time to lay in the eggs. So shaking it into an empty hive, even with drawn comb, may not allow for transmission in a package. But if you shake it and you actually shake it into a weak colony, try to boost it that already has brood, now you can complete it with package bees. So there's something to think about there. If you're using package bees, if it goes into empty equipment, you're probably safe. If it goes into, or you boost it with sealed brood, you're shooting yourself in the foot a bit. So yeah, just a point. Okay.
Jeff, I would like to confirm what Rogan and Dan reported, that we tested here in China on dead bees, and the mites survived for maximum 3 days. So more or less similar results.
So Alex, your thing was only 3 days on dead bees. Yeah. Again, I think we need more. I think humidity, somebody mentioned humidity. Humidity does play a role. These mites are very lightly sclerotized. And so I think humidity could play a key role in that. And yeah.
We used incubators. Yeah, we used incubators, and we controlled the humidity, of course, but still after 3 days, the mites were dead.
Okay. Okay. Well, good point. Again, I was surprised at the 6 days on dry comb. I was totally surprised because everything else had been 3 to 4 days and they die. Just like you're saying, Alex, when you're dead bees. I'm going to move to I had several things about control. And of course, in Europe and the Americas, we use oxalic acid a lot. And I think I put it in the chat to everyone. My oxalic acid test was with a slow release oxalic strip, and it just didn't seem to work. I know that fumigation with oxalic is being used in Thailand and Korea. So oxalic acid can work, but it ties in with the brood break. And a big thing of BeeGuards is we're promoting brood break as a natural control method for Varroa, could be used for Tropilaelaps now. Any experience with, I think Dan or Rogan, you guys did the brood break stuff. Did you try oxalic acid? I just can't remember. Tell us a little bit about, I know you use formic, but did you use oxalic?
Yeah, we added a oxalic acid dribble as after a brood break for one of our treatment groups. Saw a marginal increase in Tropilaelaps, like efficacy essentially, but it wasn't significant really. And in reality, our brood break was essentially 98-99% effective. And for our study, we're trying to see if we could get 100% efficacy to have this as a recommended thing for USDA APHIS. And so we weren't able to get that with the addition of either a formic acid application or an oxalic acid application. I would be interested in seeing oxalic acid on its own, but for the similar reasons that it's not very effective with Varroa mites with, high levels of brood, I would worry that it wouldn't be that effective for Tropilaelaps as well.
And Jeff, maybe you can fill us in on the starting level of the Tropilaelaps, because we're doing some trials with one of the extended release products called Broxan on Varroa here, and it seems to have like a marginal effect, particularly if you have high levels that are starting out. Like it'll kind of level off, but it won't drop the numbers down. So maybe you're seeing some kind of similar situation where you had pretty decent Tropilaelaps levels, and it just wasn't able to bring the mites down, but maybe it did still kind of manage them at a reasonable level.
I think for me, on oxalic, because some questions are, what are our future research things? And I think a brood break with the oxalic acid fumigation might be quite telling. And I've used dribble as well because it's easy, but fumigating with oxalic, right, at the maximum part of the brood break might work. I don't know. So I think there's still promise there.
That's what Dan and Rogan did. That's what Dan and Rogan more or less did in oxalic acid.
Yeah. I thought they used dribble. So yeah, the dribble, Yeah, dribble wouldn't get quite as dispersed as maybe as a fumigation. But yeah, that's why we do research, because we go in thinking, oh, this has got to work. And for me, like Amitraz strip, the first time I tried it, it didn't work at all in Thailand. And I think it's just a distribution problem. Any other broad thoughts on control? Where are people going with some of the control? The Auburn group has touched on some of their efforts. I know we've got active stuff going on in Georgia. Vicky, I hadn't heard from you. Are you working with, what are your thoughts on mite control at the colony level? You unmute. Yep. Unmute, Vicky. You're still muted. Yep. Vicky, you don't have the excuse that I have. It's early morning here. It's middle of the day. Can you unmute, or we'll come back to you? Okay. Anyone else on colony level control at the moment? Just any other thoughts? Again, there was a lot of, obviously beekeepers are particularly interested in that. What have I got in my toolbox to work against Tropilaelaps? Oh, there was a question about, are beekeepers using more treatments for Tropilaelaps? And this gets to something that just Jeff and the other Jeff have discussed. In general, in Thailand, and this was shown in some of the nice graphs that you showed about the buildup of Tropilaelaps mites. In my experience has been in Thailand, they treat more for Tropilaelaps and treat more often, whereas in Korea, they focus on Varroa. So there's a difference in climate, and we don't understand that. But maybe Auburn just touch on that briefly. I like that figure where the mites are really increasing in Thailand just more rapidly than in Korea. That was a beautifully displayed example of that.
I'm going to turn that over to Dan, but I'm going to, in a second, I'm going to dig up a URL link to the PAM, one of the PAM videos, because there they did a couple of case studies of the beekeepers in Thailand to highlight what their kind of seasonal mite management schemes kind of look like. So I'm going to dig that up and put it in the chat for everyone. And then I'll turn over to Dan to talk through his graphs.
Yeah. Yes, so based on following the Tropilaelaps mite populations in our untreated control colonies in a couple trials, and then also the Varroa levels, we can see this dramatic difference in how many fold increase you see for Tropilaelaps versus just a small increase in Varroa. And yeah, so it's somewhere on the scale of, if you see a doubling or a quadrupling of Varroa mite populations over a couple months, I believe we were seeing a 10-fold increase or more in the Tropilaelaps populations over 60 days. So unless you really effectively suppress your Tropilaelaps populations, like perhaps winter can do, or in an area with severe winters, or a complete brood break may be able to do, that could really bring down those Tropilaelaps levels really far, and then that could give you more time. But if you don't suppress your Tropilaelaps populations sufficiently, then I would expect that more frequent treatment is going to be needed.
Yeah. Thanks, Dan. Another question that came in, and we've already touched on this, was how do mites survive broodless winters? And we've already touched on that quite a bit. I mean, there's at least several ideas out there that how they could do that. But I think the one that's overlooked the most is that most of our colonies may not be going completely broodless in the winter. I did some quick sampling in Korea in February, and I sampled like 50 colonies, and only in one could I detect a Tropilaelaps, uncapping 300 cells. So they were there, but at super low levels. And then there's, of course, all these alternate host ideas. So for some reason in the winter, it's seasonal. The mite levels do go down. What they're doing to survive, we don't totally understand. But they're back in the spring. They come back in the spring, and they're there. And could be reinvasion from wild colonies in the area or whatever. But yeah, they're always there.
I'm again in the
I'm sorry, go ahead. Victoria, yeah.
Yeah,
Is
I'm a
that you,
bit,
Victoria?
yeah, it's me. No.
Yeah.
Do you hear me?
Yes, Vicky. Yeah, we hear you.
Yeah, what I wanted to say that I am wondering, I mean, it looks like that if you have a broodless period, then it's supposed to help the control Tropilaelaps. Even if we do it artificially, if we cage the queen for several months, it should work, shouldn't it?
It should work. Yeah, a nice brood break should work, and maybe we even with the brood break, maybe we need some application of an acid or something. Who knows? But yes,
yeah,
yeah, brood breaks, and that's a big part of the BeeGuards.
Yeah, I know. I'm a part of it. I'm a part of it. So that's why I really, it surprises me that what I understood when they did Dan and sorry that I don't recall or remember the names
Dan and
of
Rogan.
the other.
Yeah, Dan and Rogan.
And the Rogan, when they did the caging and the trickling of oxalic acid, it didn't work. I mean, how long did they cage the queen?
Rogan, step in there and comment on Vicky's.
Yeah.
So we caged the queen for 24 days, trying to get a full brood break, and that was because if the queen had laid a drone egg, you know, the day that we caged her, then theoretically it should have that drone should have emerged, you know, by day 24, and so there should have been no brood left in the colony.
And it didn't work. We caged in BeeGuards for 25 days.
It did work.
Yeah.
I mean, from a management perspective, Vicky, it did work. I mean, it was, I think Rogan mentioned like 98%.
But why not 100?
Effective.
But how come not 100?
Yeah, so we did a follow-up experiment. One of the shortfalls of a lot of our Varroa management work is that we have our control colonies in the same yard. So we instantly thought, okay, getting
you got
some
the reinfestation.
Maybe. But then Dan and Rogan did a follow-up trial where we maintained multiple yards with multiple sets of different treatments. Like one yard just had controls, another yard had just controls, another yard had brood break. And we still got quite similar results. So that was a little bit puzzling why we didn't get exactly 100%, kill rate. Maybe there's still some drifting. I mean, there could be other populations of bees in the area or things. So that's something that we're still investigating, because of course it only takes like one mite or two mites to survive, right? So from a beekeeper management point of view, brood break seems to be perfect, but from maybe eradication point of view, that's where we're kind of still scratching our heads over a little bit.
So maybe.
Go ahead, Vicky. Yeah.
Sorry. So I'm saying, so maybe the alternative host is still the other possibility, because that's what enables them to survive, even if it's broodless, so they go somewhere else. But what I would like really to raise the issue that I think we didn't talk about is what actually happening in Dorsata, where they're its original host. How do they survive there? Because probably they also, what do we know about their behavior and survival in Dorsata bees?
So I can tell you, I even question that it's the original host, because Dorsata migrates. If it migrates, it has to set up new brood. How's Tropilaelaps getting there? I don't know. So I think there's if somebody, that's a great research project, and we just don't know enough. We know that Dorsata migrates and has to build new comb and lay. So it's outside of the survival range of Tropilaelaps if they don't feed on adult bees. So we need to, if we understood that a bit more, maybe we'd understand how it's surviving in European colonies, European honeybee colonies. So we don't know that. I just want to point out a quick thing between what this discussion for the broader audience. What Jeff and Rogan and Dan were trying to do was eliminate Tropilaelaps from colony, make them mite-free. And the answer to that is it's biology. Nothing is ever simple in biology. So, but I mean, so Vicky, for control for the beekeeper, it's an effective tool. It reduces the mite levels down really low, They were trying to eliminate it, so eradicate it from an area. And that's quite a different thing. That's the difference between surveillance, they're just finding the mite, positive or negative, versus quantifying it. So there's always a difference there. One more question I'm going to add, because it kind of gets to the point of why we're having this seminar. It says, since the discovery in Georgia has it spread, and a very brief talk about bridges. We've talked about mites, we need to talk about bridges. And what are those bridges? Those bridges to Europe, more Europe, are Turkey and Turkey, Ukraine, Belarus. If you look at a map and you look at where we think it is and where it's moving, Ukraine, Belarus, and Turkey, that we need to be doing a lot of sampling there, because that's in Turkey, they migrate heavily across Turkey to get to pine honey. So I know there's sampling going on, but it also gets to this reporting issue. When we find it, we need to at least report it artificially within the research community, so we're more aware. But there's a lot of need for what we're doing here, which is raising awareness and working on surveillance tools. I think we're getting close, and I'd like to stop the question and answer. We've answered most of them, or tried to answer them, and I'd like to go around the table and just have some quick summary thoughts. So I'm going to start with Jeff, because I'd already warned Jeff, and I warned everybody else in the thing. Think about just what's your final thoughts on Tropilaelaps, where we are, where we're going, what you'd like to see done. Jeff, go.
Thanks, Jeff. I guess I want to take the route of continuing just like the collaborations and the funding initiatives. The mite is spreading. It'll probably continue to spread. So I really hope that all of our initial efforts right now just doesn't kind of fizzle out, and then in 3 years' time we're back to starting this all up again. So that's kind of one of my goals, is to really work towards snowballing our efforts here. Of course, we don't want to take our eye away from the major stressors in each one of our areas, but I think we should really just try to keep this going. For example, have a CLOS task force. I definitely want to get in the room with the panelists here and just have some discussions kind of moving forward, and just see how we can keep this momentum going. Yeah, just a quick. I'm gonna close with Raquel, Giles, and then Cecilia. Let them have the last word as kind of summing it up. But I'm looking down. I'm going to go to Alex and then Rogan. So Alex, go. It's on my screen. That's the way I'm organizing it. Yeah.
Yeah, no problem. From my point of view, I think that we have to improve our knowledge and understanding about the mite under temperate climatic conditions, since most of the data comes from tropics. Yeah. So that's why we have a good chance, and that's why our team, within the frame of the BeeGuards project, here in China and also in Georgia, we are investing a lot of energy on two main aspects. First are the biological features and biological aspects, to understand the dynamic within the colony under these temperate conditions. And second, we are addressing more pragmatic, more practical aspects about the control. But when we discuss about the control, we should not ignore Varroa mites, since Varroa is also within the equation here. And we have to think on the methods and concepts how to address both mites in our beekeeping operations.
Yeah,
so
I would agree
these
with
are
the
my
both-sided
thoughts.
approach. Yeah, thanks, Alex. Rogan, and then I'll go to Dan. We do an Auburn thing back to back. Rogan, then Dan.
Yeah, I'll just reiterate some of what Jeff said. I think we need to continue to work together on this and really work to have that great monitoring and detection. I think we need to be continuing to say where the mite is at and keep things most up to date and have a really standardized procedure for doing that, because as this mite continues to spread, there's going to be a lot of different reports out there, and so having that standardized procedure is going to be really important for knowing where it's at.
Good point, Dan.
Yeah, and I would follow up Rogan's point by saying that we're making some travel plans to visit some additional countries the next year, and hopefully to get a little more clarity on where it's spreading to. From a like research needs standpoint, I think Alex's point about needing to understand how Tropilaelaps is going to, what its biology is like in temperate areas is really important. That also connects to some beekeeper management that's used in, for example, the large-scale beekeeping in the US, whether indoor cold storage can be a useful tool against Tropilaelaps. And then from a biology and from a perspective of understanding the host parasite dynamics, when we get Tropilaelaps and Varroa at the same time, we need to start thinking about thresholds for both of those mites and how to do integrated Tropilaelaps and Varroa management. So that's I could see that as quite a challenge to overcome. And then we're working on early detection techniques, including DNA-based detection. So I hope that we will have those results out quite soon. And then finally, we're going to keep on working on effective management techniques.
Thanks, Dan. I'm going to go to Iraqi, Dan, and then Vicky. So sorry, I didn't give you a warning, but go ahead. Yeah, from Georgia.
Okay. I think since we are addressing beekeepers right now, I have to admit that it's paradoxical how we digest information. Actually, beekeepers tend to believe some tiny possibilities of survival mite on alternative host and things like that, rather than focus on what is known, well known, that mite is dying from hunger. And in Georgia, it's very, very evident. And beekeepers quickly learned that synthetic acaricides will not work in original shape like we were designed for Varroa. And we started to make it at home. I mean, making some amitraz strips and overdosing. So it raises new issue of food safety. And I think perception of beekeepers and how to address them is very, very important aspect once mite is established in somebody's country. So that's main concern for me right now.
Okay, yeah, very good thoughts. And on that control note, I've seen things used in Thailand and even in Korea that we would never use in other areas. They would never be approved. So yeah, it's important that we work to get those things approved and have them for the tools for the beekeeper. So I think Dan and then Vicky. Dan, if you're there.
Who is Dan now and then me?
It was Dan and then Vicky, but I'm not hearing Dan. So Dan may.
No,
Dan
I'm here, Jeff.
Oh, there you go.
Okay.
Okay, Dan, go.
Yeah.
Yeah.
So I think my first thoughts are to get the distribution maps updated and all these. We've got lots of anecdotal evidence of that mites are here and there in different regions, and I just think probably that would be a huge help to get that updated and confirmed. And I think that Dan's already mentioned about his travel plans to try and get some unknown areas updated and just to find out where this mite is, and then obviously I think the stakeholder engagement is also a huge thing. We've been working on that a lot at the National Bee Unit, and I think the profile of Tropilaelaps has gained huge momentum in the last year or two. So I've had all sorts of members of the public, beekeepers, all sorts of things mentioning Tropilaelaps now, whereas a year or two ago they didn't even know what the word was. So yeah, and I think understanding, you've already mentioned the mites in temperate climates, but just also in the way that Western honeybees are managed in our management systems, the sizes of the colonies, some of the amount of brood, as we've worked a bit in northern Thailand, and just the sheer volume of brood that we produce in the West with our short window of opportunity to do so, and particularly in North America, probably over the UK, but we can produce a heck of a lot of brood in short space of time, and I think that understanding how the mite does develop under those circumstances is probably a big question mark for me anyway. So I think that's probably all I've got to say on that, Jeff. Thanks.
No, good thoughts. And in fact, I mean, most of our work in Thailand and in Korea, we're working with smaller colonies, 5 to 7 frames of brood. So yeah, the colonies in Europe and Asia, I mean, Europe and the Americas would be much larger. Okay, Vicky.
So I just came back from a trip to Azerbaijan, Kazakhstan, in Kyrgyzstan, in search of and met a lot of beekeepers. And so the issue I didn't see at that moment, although I saw in Kazakhstan in November Tropilaelaps, but now we didn't see any. And it raises the question, where did they go? Are they gone or not? Will they reappear? So I will go there again, I guess in September, to see what's going on. Anyway, I can tell you that beekeepers that I talk to are aware. We gave, me and Anna Brandov gave talks to them about the Tropilaelaps, so they're aware. But the Varroa problem there is terrible. Some of them doing 20 treatments a year. And they lose like 70% of colonies, some 50% of colonies, because they have a very bad situation with probably with resistance to all kinds of pesticides. So as some people before, Alex and others, raised the issue of integrated management, yes, we will need to have an integrated mite management, because even if amitraz is very useful, or fluvalinide will be useful against tropy, but it would not be useful for Varroa, then we will end up in a very, very, very big problem. So I think as Varroa, it's not that we are going to have Tropilaelaps instead of Varroa. We have both. And it's really a very big problem. I have contacts with people Russian-speaking world, and as far as I know, in Belarus they don't have Tropilaelaps in the moment. At least they have not detected any. Regarding other countries, there are situations which are quite problematic, and I even discussed it with the Deputy Minister of Agriculture of Kyrgyzstan, who happened to be a beekeeper. And I realized that the thing is that the traffic of packaged bees between the countries is a very big issue. It's a very big economic issue. And it will be very difficult to achieve the situation that the countries will admit that they have Tropilaelaps. And if they don't admit, I don't know what we can do. We can just hope that some local beekeepers or local people, like Irakli in this situation, will come up to us and report.
Yeah, good point, Vicky. I think Iraqi covered that. Sometimes there's political issues and pressure not to report, and we should try to find ways to overcome
exactly.
that. Yeah.
It's a very, very big problem.
Yeah. Agreed. If we're going to slow.
And one more point is that now a lot of beekeepers sending pictures with all kinds of creatures that are not Tropilaelaps. But we also mention it to them that it's okay. We are ready to get, all these. We should also be aware that some of the, information might be not really Tropilaelaps. So we should try to get the samples and analyze them properly.
Yeah. But because we're getting many pictures, it's a good thing because we've raised awareness.
Exactly. That's very good.
Yeah, it's important. I'm going to close my remarks really quickly and then turn it to Raquel, Giles, and Cecilia to have the last word. I'd like to thank Auburn University for helping to host this, and BeeGuards obviously for putting it on, and Apimondia, the organization that I represent. Just a couple of quick thoughts. One is watch the videos, become aware, because as a beekeeper or an inspector, you're our first eyes out there to be aware of what might be going on. Whether you report it through WOA or through your local bee inspector, do report what you find. Lastly, I will point out that we are hoping with Alex and others in the BeeGuard team to hold a hands-on workshop in maybe this summer, maybe into next year, but so that people can come and see it firsthand. And we'll look for that later as we make plans for that. So from my standpoint, I thank the speakers because a lot of work went on to put together these excellent presentations, and a lot of research went on behind it. So thank you all as speakers. So, and now I'd like to turn it to my co-host and technical person for the events, Raquel, who's help us put all this together, keep us on track. Raquel, do you want to at least say hello or a final word?
Hello. Yes, I want to thank you all the speakers as well, because I'm also a bee scientist, or I used to do more research, but not in pests, more in nutrition. And I've been learning quite a lot about this, which is also important, and also try to promote these within the beekeeping community in Portugal. And I had just a note for the wrapping up. I think it's important that we keep promoting these kind of workshops to share this knowledge, forefront knowledge, and also to attend these workshops and keep up the critical thinking and be observant, because there won't be a one solution fits it all. So thank you all for attending and being patient with all the technicalities until now. So thank you.
Thank you, Raquel. Giles, our
Yeah,
work package leader.
Yeah, just to find a few final thoughts from me. I mean, it comes down to biology, doesn't it? And understanding the biology better, not just of the mites, but also of our bees. The bees that we think we know very well, perhaps the climate's changing and their ability to raise brood in the winter's changing, and therefore the risk of Tropilaelaps is changing. So there's some really good observational biology that needs to be done. Best that we do that together as a community. There's some really good examples of that in the presentations that have come along. Working together as scientists, I think we can present a united front and a way forwards for beekeepers. And then the second thought I wanted to share really was this concept of how any beekeeper watching this can make a difference. You know, by watching the videos that we've provided, the information that we've supplied, you can make a difference by coming back to that point that Victoria made very eloquently in one of her slides, where if you catch these invasions early, then we can do something about it. An example, in the UK, we're an island. We're doing some really good things with the yellow-legged hornet at the moment, keeping those populations down. But equally, we've got examples of where we got it wrong with Varroa. It came in 1992, and some of those very first colonies that were reported had nearly 20,000 mites in them. We know Varroa doesn't do that in the season. It's been there for years. So watching this, staying on the ball and monitoring are all really, really crucial steps for us to make a difference. And so thank you to everybody for your contributions today. It's been a really useful webinar.
Yeah, and thank you, Giles, for that comment or the comments. And yeah, Vicky's thing about early education was critical. The other point I want to make is that this webinar will be available long-term. We'll post it on BeeGuards, we'll post it on Apimondia and other sites. So the work won't, and there's so much good information here that people can refer to it. So now, Cecilia, our global BeeGuards thing, I'll let you have the last word, and thank you all for help with this. Yeah. Cecilia.
Thanks, Jeff. Thanks, Jeff. Well, thank you to all the speakers, really, well, for your research, for your work, and for presenting here today. Thanks again to Auburn for hosting us, and thanks to Raquel and Jeff for the organization behind this. I want to say I'm really also proud and happy of the work we're doing within BeeGuards, because we had planned it all before even before we started being afraid of Tropilaelaps arriving. But still, we did have a plan of supporting the beekeeping sector and being prepared, which I think is exactly what we're doing with this webinar today, and with the other activities which we have planned, like the workshop that you were mentioning, Jeff. And also, I think it was highlighted today by several talks that the brood breaks, what you call in America the cultural control, and we call the brood break, how effective this can be against Tropilaelaps. And we have as aim within BeeGuards to promote this kind of method to fight and control Varroa, and to support the development of resistance against Varroa mites within honeybees. But we can see it's so effective also against Tropilaelaps. So really, if you all go also have a look at the BeeGuards website, there was one of the questions which was somebody was asking how to do the brood break. So still, it's such a simple beekeeping practice, but still not well known. And I can see even within my country, in Italy, where it is quite widespread, still there are some parts where beekeepers are not using it, are not familiar. So really, it is something where I think we can really still do a lot, and we can see that it's really, really important to do it because of the risk of Tropilaelaps. So I won't continue further with this. Really good that the whole webinar will be online, and thank you for doing this. It will be a precious resource, I think. So thank you, everyone. I think this was really good. Bye-bye.
And we'll sign off now. So thank you all. This was wonderful. Yep.
Bye.
Bye for now.
Bye.